5, 4, 3, 2, and 1.
Okay, it looks like we have a crowd gathering on YouTube. And I hope everyone was able to find the correct link for the stream today. Got to get all my windows here. All right, so definitely, Trisha, I cannot see the chat, so I'm gonna need someone to help me with that. Jose, yep, maybe you could mute. That's good. All right, it's 10 o'clock. My name's Lena Ting. I'm a professor of biomedical engineering at Emory University and Georgia Institute of Technology. And I'd like to welcome you to this Emory Theory and Modeling of Living Systems virtual workshop on human and animal movement. Where does stereotypy end and variability start? We have a very exciting lineup of speakers, and this workshop, in this workshop, we have 10 minutes of speaking for each speaker, followed by 10 minutes for discussion. So I'm hoping that you will be actively discussing in YouTube. We have helpers monitoring the chat stream, and those questions will be funneled to our panelists. If you're one to tweet, I have our handles down here for the Emory TMLS initiative and the Georgia Tech and Emory Neural Engineering Centers that are hosting this event. And our hashtag here is TMLS Movement Workshop. At the end of the session today, we will also have a 20-minute panel discussion amongst everyone. So if you have broader questions, we will hopefully be tackling those at the end of the workshop. Now, before we begin, I want to acknowledge that we have gotten funding from the Emory Woodruff Health Sciences Center and the Emory College of Arts and Sciences to host this workshop. And it absolutely could not have happened without the help of and dedication of Amanda Johnson-Scott and Tierra Ward, who are making sure that all the emails and live streaming happens. I want to acknowledge the my co-organizer, Tricia Kazar from Emory Division of Physical Therapy, and our consultants in Emory Physics and Biology, Ilya Nemenman and Gordon Berman. And behind the scenes, we have the excellent support of, these are the people monitoring the YouTube channel, several PhD students across Georgia Tech and Emory, Chase Rock, Megnan Wu, and Laksh Punith, as well as assistant professors Jing Xu from the University of Georgia and Mark Lyle from Emory University. Now, it's been a very, very rough year, and we originally wanted to have this workshop in person almost exactly 1 year ago, in which case, hopefully all of you would have been here with us in Atlanta, which unfortunately was the site of horrific crimes against Asian Americans and women in particular. And being an Asian American woman here in Atlanta, this hits really close to home, that that we all need to work together to make our scientific and personal lives safe for all of us, to allow everyone to participate. So I wanted to have people reflect on that and hopefully provide some resources to help people move forward in the sort of work to embrace diversity, inclusion, and anti-racism. I think it's particularly appropriate for this workshop, since we are really embracing diversity at its core as a topic of study in scientific inquiry. And thanks to here are some resources provided by Asian Americans Advancing Justice on combating violence against and anti-racism against Asian Americans. I also want to share with you some resources for bystander intervention. I personally need help putting, my intentions into actions in the moment, and that can help everyone. I also want to stand in solidarity with our colleagues, who are Black scientists, and point to a couple papers authored by my colleagues here at Emory and Georgia Tech that were published in Nature and Cell journals. And finally, there are little steps we can all take just to make our environments more welcoming and inclusive. And I have a pretty old document on my website that needs to be updated, but you know, thinking about how we interact with people in our everyday lives in the lab and beyond. All right, so with that, I'm going to now introduce, I'm going to take my stop sharing and let Monica put her screen up. And our first speaker today is Monica Daly, who is an associate professor of the University of California, Irvine. Take it away, Monica.
Good morning, everyone, and thanks, Lena, for that introduction and for really focusing on some of the most important issues of the day, too, that we all need to work together to combat racism and promote inclusivity in all the professional activities that we engage in. And I think that's really important. So I'd like to start today by talking about the question that was posed by the workshop, where does stereotypy end and variability start? And to do that, I'd like to introduce you to Greg the guinea fowl. This is a slightly old video that was put together by my former postdoc, Yvonne Bloom. And what I'd like you to notice is to watch as this bird enters into the runway arena from the right side of the screen and runs across the platform. And each time this is a different trial, and it's a sequence of multiple trials stitched together. And one of the reasons we work on guinea fowl is because they're athletic bipedal animals that we have always considered to exhibit very stereotyped behavior. So it's an animal model that allows us to study bipedal locomotion and some of the fundamental mechanical demands of bipedal locomotion and issues of stability in an animal where we can get them to exhibit relatively consistent behaviors and then expose them to terrain perturbations and look at how they respond to these terrain perturbations. And, you know, so this bird is running fairly consistently. And one of the behaviors that is fairly stereotyped is if we look at the leg angular cycling rates in these birds, they exhibit a very consistent leg angular cycling pattern as a function of speed during locomotion. And this is consistent even when they encounter fairly large terrain perturbations, that the leg angular cycling remains consistent and is adjusted only over relatively long timescales. However, we also see some big sources of variability. And one of the challenges throughout my research has been trying to understand and make sense of the sources of variability. Some of our early work really highlighted the variability. We did studies of exposing the guinea fowl to unexpected terrain perturbations and looked at the mechanical responses and how they avoided stumbles and falls. And the birds showed a wide range of responses in terms of the mechanical energy response of the center of mass. They were very consistent in being able to maintain forward speed and avoid falls, but there was a high degree of variability in the energy responses and the loading in response to these perturbations. However, there were challenges in making sense of this variability because of the limited sample size. So we had only 39 trials where we could expose these birds to this unexpected perturbation. And this was across 5 individuals in 3 conditions. And we had to kind of divide it into 3 response mode categories to try to make sense of the variation in the mechanical response. So we really had a limited statistical power and ability to make sense of the sources of variability in these data sets. One of the sources of variability might be individual variation, but we just didn't have the number of trials and the repetitions within individuals to make sense of that variability. So that's a question that's been kind of lingering throughout our research over the years, is trying to make sense of the sources of the variability. And one of the things that we've been thinking about a lot are the potential sources of variability that arise from animals trying to balance multiple task demands in complex behaviors. So we are studying non-steady locomotion, and there's multiple possible task level priorities that animals must consider and prioritize: desired velocity, maneuverability, stability, disturbance rejection, and potentially considerations like force limits for injury avoidance and energy cost. And one of the challenges is that we don't know before we actually make the measurements and do the experiments which of these are really going to take priority for certain kinds of non-steady tasks that we haven't studied before. So thinking about some of these issues, we decided to design an experiment where we were a little bit more explicitly putting some of these potential task level priorities in competition directly with each other. So one of my former students, Hannah Goldsmith, designed an experiment on turning locomotion. And this was a series of 4 terrain conditions. On the top left, we had just a control straight runway with regular high friction terrain as a reference condition. And then the top right, the birds encountered slippery terrain with the coefficient of friction similar to ice. And then we had 2 turning runways, a high friction turning runway with a 90-degree turn. The birds run from a start box to an end box. They can choose their path and speed to navigate this turn. And then finally, a turn condition with this slippery substrate. So our prediction is that in these turning maneuvers, we would expect the slippery terrain to force a greater priority for slip and fall avoidance. And so turning requires horizontal forces to redirect the velocity of the center of mass. But when these birds encounter slippery terrain, we expect that they will then have to slow down and take shorter steps to maintain a higher ratio of vertical to horizontal forces to avoid slips and falls. So what we expected is that the birds would slow down much more dramatically in this slippery turning condition. So we made measurements in 7 guinea fowl, and in this case, we recorded at least 10 trials per condition until the birds were unwilling to cooperate any further, basically. And we recorded every single trial in this case so that we had a large data set that we could potentially look at individual variation and learning effects in response to these 4 different terrain conditions. Alongside this, and separately at the time, my PhD student, Jade Hall, was conducting a series of behavior experiments on our flock of guinea fowl. She was interested in affect expression and personality expression in the guinea fowl, which has really not been studied extensively in the species before. And she did a series of tests to measure the propensity for risk-taking and exploration behaviors in these birds. And this is just showing the results of one of the many tests she conducted. This was called the emergence test, where the bird is in an arena and it's put in a box. The door to the box is open, and you measure how long it takes for the bird to emerge from the box. And so this is considered a test for risk-taking in these birds. So some of the birds immediately left the box. So they have a emergence time around 0. Some of the birds basically never left the box until the time limit of 15 minutes at the end of the experiment. So we have a range of behaviors in these birds. We did a range of tests, and we had some consistent clustering of bold individuals and shy individuals, and then some who were in between. So getting back to our turning locomotion experiment, and it will tie into the personality in just a moment. This is just a video example of the guinea fowl navigating our high friction turning condition. The panel in the middle is an overhead view of the center of pressure, and then the bottom panel are the ground reaction forces as it runs along and approaches the turn. So we're interested in how fast the birds approached the turn in these different conditions. Here's an example of the same bird in the slippery terrain. If you look carefully, you can see that it starts to slip and then slow down as it approaches the turn. And in this case, it does slip a little bit, and there might be a little bit of body contact. So in these birds, we measured ground reaction forces and body center of mass dynamics as they approach the turn. And we also looked at whether they fell in these turns, which was defined as a body that was not the legs contacting the substrate at any point in the trial. And this bird fell in about 55% of the trials, so there is a high risk of falls in these turning maneuvers. What's interesting is that we did see a high amount of individual variation in how the birds navigated this turning terrain. So this is an example of one of our birds who took a much more cautious approach to the turns, always came in slowly with a relatively careful walking gait, and this bird never fell in any of the trials. So the major response was that we did find that they slowed down in turns, but surprisingly they did not slow down substantially more in the slippery terrain compared to the high friction terrain. And what we find is that these birds, if we looked at the effects of terrain compared to the individual variation, so on the left, we have the fixed terrain effects in terms of how much the birds slowed down in the various terrain conditions. They slowed down a little bit more in the slippery terrain, but mainly the birds narrowed the speed range to something that corresponded to a grounded run. We saw a high degree of individual variation in speed in approaching these turns. And the speed clustered based on the bold, shy personality scores that we observed in Jade Hall's experiments. So there are some birds who are consistently bold and they consistently go faster across all terrain conditions, and some birds who are consistently shy, and they also were the ones who also exhibited no falling. And so what we're learning from this, there's not a perfect correlation between the individual effect variation in our locomotor experiments and personality, but it is certainly a factor that arises and maybe is something that we need to consider. And looking back at our past research, one thing that we have to realize is that we often pick the birds who consistently perform in these laboratory conditions we put them in. And that means that we might have biased many of our experience towards these bold individuals who will run and do the thing you ask them to do no matter what. So it's something that we're thinking about how we should address in the future. Guinea fowl do reduce speed for turning, but not much more in slippery terrain despite high fall risk. The birds who don't care about falling don't change their behavior to avoid falls, which is interesting. But there was high individual variation in speed and fall risk tolerance, which was similar in magnitude to the actual terrain effects we were trying to measure. So that's getting us to thinking about how we should deal with the influence of personality, including things like bold, shy personality and risk-taking in biomechanics research. But one reason for hope is that speed is a large predictor of the variance in many of the biomechanical variables, both within and in between individuals. So if we can think about looking at that variance as preferences for specific speeds according to risk tolerance, then we might be able to get somewhere with that. Great. And with that, I will take questions if we have time.
We definitely have time for questions. Thank you for that, Monica. And one of the questions we got in the chat was, how do we know if the particular birds are choosing the gate strategy? And are there, other biomechanical or cognitive constraints that might have them switch strategies? For example, I was thinking, I study falls, and we always say that the cost of falls is high, but clearly there isn't really a penalty here. So what if they're like spikes on the ground? Would you see changes in behaviors?
Yeah, I think one thing we need to look into more is thinking about looking at changes in behavior over time in response to specific experiences in specific events. So perhaps the birds change their behavior once they've experienced a fall that had some consequence. But as you said, it's clear that for the birds who fell, there didn't appear to be a real negative consequence for them. And so they continued to behave in mainly the same way. One thing I'll note is that the boldest bird, the first time experienced the slippery terrain, it actually collided with the far wall. And so it did slow down just enough to avoid that in subsequent trials. So there's certain...
Oh, that's good. I haven't learned to do that yet on my snowboard. I'm definitely a person who's prone to injuring myself, and apparently I haven't learned from that. So I definitely resemble that guinea fowl.
I'm definitely one of the shy guinea fowl. So
So there was a question about, can you perturb their personalities with like anti-anxiety drugs and see if that changes anything?
oh, that's a cool question. I think one of the questions we have to address is that its personality is part of the story, but we also, and so maybe anti-anxiety drugs would be something to try, but it might also be that these individuals are correctly assessing their own individual risk. It might be that the individuals who are stronger are behaving in a bolder way because they know they have the capacity to withstand larger forces.
Mm-hmm.
And so we don't really know what the interaction is between sort of like a general robustness or vigor and personality here.
Right, or maybe just pain tolerance.
Yeah.
I actually think I have a high pain threshold. I guess one of the questions is whether there is something that you can see in their normal walking, such as variability in their movement or ground reaction force, that might predict what their behavior would be in the unstable conditions.
Well, that's exactly one of the things that Jade Hall was looking into was locomotor-based indicators of personality and mood in these birds. And what we find is that mainly what we find is that individual birds do behave very similarly across a broad range of contexts. The birds who are careful and cautious and risk averse are always careful and cautious and risk averse. So I do think that we can characterize their tendency to be risk avoidant across different experimental conditions based on locomotor measures.
The question was like, are these personalities clustered around other variables such as age, sex, or size? Maybe the bolder birds are,
Yes, absolutely, they are. It's complicated because there's multiple factors, but the bold birds do tend to be male birds. However, there tends to be a boldest female in the flock, depending on who is the current dominant female in the hierarchy. So there's a little bit of changing of personality expression over time as well, based on the flock hierarchy.
Oh, that's interesting. And does that, I know in some groups, right? So the female who is the lead can change, and then that actually changes quite a bit about even the physiology of those animals. Is that?
Yeah, absolutely. And those change over time with seasonality. So,
Oh, wow.
in breeding season and things like that. So it becomes very complicated to try to really address all of these factors at once.
Yeah, I definitely think that that's one of the main issues here, is that there are so many factors that can affect variability. And here you very nicely kind of isolated a particular one by manipulating, developing an experimental condition to do that. And I really like the fact that you're moving away from just steady state, kind of straight line walking, which is, what many of us do, and which is traditionally what has been studied. So the richness that you get from moving away from that is great. I have, not sure I understand. So you did say that the one that ran into the wall then stopped doing it. So maybe slightly modified their strategies.
Yeah, there are some learning effects. We're still kind of working out coming up with the right approach to the statistical analysis, but in our preliminary analysis, it does appear that there are significant learning effects, although they're smaller than we would have anticipated based on how frequent the falls are.
Are there other personality axes that you can test that's beyond bold shy that might also play a role here?
We
like anxiety? I don't know if that's one.
I think that they're kind of closely linked. The anxiety, the bold shy I think it's not really the perfect name for what is being characterized there, because it's really a risk-taking propensity, but it's influenced by the physiology of stress response. So I think that a large factor is that shyer individuals are more sensitive to stress responses and take longer to recover from stressful events compared to bold individuals. And the origin of that and how much that changes over time, depending on current physiological state, is kind of a complex question that we don't have all the answers to for guinea fowl in particular, because it hasn't been studied extensively.
Great. Thanks so much. We have a lot of interesting questions in the chat. So maybe if you could, we can follow up during
I'll
the
do that.
session. Matt Tresh says, very nice work. Bold shy might be an interesting context to approach motor control strategies for recovery from an injury as well. And I think that makes a really nice link.
Yeah, I think that's a great point.
Yep. Great. All right. Well, with that, we're going to move to our next speaker. Is Mackenzie Mathis. Go ahead and share your screen. Mackenzie we're grateful that she's joining us from École Polytechnique Fédérale de Lausanne in Switzerland today. All right. Thank you, Mackenzie.
Yeah, thanks, Lena, and thanks to the organizers for having me. And I really appreciate your introduction to this workshop. I think there's never a bad time to think about our impact on the world. So I really appreciate your statement and certainly support everything you said. So today I'm going to talk about how maybe new tools can help us study variability. So I'm not going to talk too much about original data on this, but more or less give you hopefully some thoughts and ideas and potentially new tools to add into your toolkits as you study these types of things. All right, so as many of you know, the study of measuring movement is maybe as old as mankind, but certainly documented nicely by Edward Muybridge and others hundreds of years ago, where they actually came with a scientific question to ask about whether a horse could lift all four of its legs, or if it does lift all four of its legs off the ground at the same time. And so they set up this quite elaborate camera trap in order to take photographs, which they could concatenate together to make a high-speed video. Of course, now with modern high-speed video cameras, we can actually even see a phenomenon in movement that we've never been able to see before. So here's a really beautiful example of these songbirds that sing, and they have this quite elaborate dance that they do to potentially attract a mate. And so this is actually faster than what we could see by eye. So I think this is slowed down by about 1,000 times actually, but it's really revealing new things about movement. And shamelessly, of course, I can show my own video, but I think many others, of course, become before me. But using videography has become a really powerful workhorse in modern neuroscience and biology to study movement, just like we saw with Greg the guinea fowl. I'll have to swap this out for Greg, Monica. That's such a beautiful example. All right, and so for many studies, this field of marker-based pose estimation being the geometric configuration of the limbs as you define them by key points, is really quite robust and has a nice long history. So for example, I think most of you recognize this scene where you have a human, they're in a room and they wear a lot of markers. I'm not sure if Greg was wearing markers maybe, but nonetheless, here in humans, you put these on and this becomes a much easier computer vision challenge, right? You can simply segment these bright reflective dots and you can get motion from this. This has been well documented, just the fact of this motion actually from say Johansson and others can give a lot of information, and even David Marr, about the characteristics of movement, so just the skeleton itself. Putting markers on animals is, I would say, significantly more challenging. They're certainly less compliant than humans that are willing to get in front of a green screen. I really love this paper from Carlos Vargas Irwin when he was with John Donahue and collaborated with Michael Black to put these markers on these monkeys while they did 3D reaches. And this work really took, talking to Carlos, it was a huge amount of effort actually just to train the monkey to accept them to wear these markers while they did these movements. And so a lot of the bulk of this early work to train these primates was just literally this. So months and months of work, training them to accept these markers before you could even get to the data acquisition phase. Of course, there's been really elegant work in smaller animals, like this nice example from Ayman Azim from the Jessel Lab several years ago now, where they looked at kinematics and variability, and in particular when they perturb some of the spinal circuits to show that there can be deficits with this. So I think this is a really powerful approach, but it does have its limitations like all methods do. So starting around the end of 2014, deep learning really burst onto the scene of human pose estimation to do this markerlessly. And so this is quite a nice advantage because not only can you go back and look at old videos, you a priori don't have to define the key points by putting markers on them, but it really opens up the toolbox of what you can potentially study. So here's an example from a nice algorithm called OpenPose. And as you can see, arguably, these are humans in the wild dancing on a pier in Sydney, and this is quite impressive performance. And so if any of you are new to this field of deep learning, the principle of it is actually quite simple. The idea is that you have an image, you have a predictor, namely a deep neural network, and the idea is to predict a pose right from this. The idea, though, of course, is that you need to train this predictor. So you get ground truth annotated images, you train this predictor, and this often needs a lot of required labeled data from humans. But this is a massively growing field, and it's quite impressive developments in computer vision are still happening every day. All right, so about 3 years ago or so, 3.5 years ago, myself and Alexander Mathis were really wondering if we could transfer this to the laboratory. Namely, could we do this in an efficient way with way less data? So the human pose estimation algorithms are typically developed on these very large benchmarks with tens of thousands of frames, which didn't seem quite practical for most lab applications and might not even be necessary for most lab applications. So I'm not going to go through the details, of course, in this talk, but suffice it to say what we found is that this notion of transfer learning, i.e. pre-training our networks on other tasks like large-scale object recognition tasks first, and then fine-tuning end-to-end these backbones, we could predict key points, human-applied key points, virtually, with very, very little input data. So this is an example of a mouse that had not been trained on this identical mouse, but certainly a similar setting, and very few training images were used actually to get this performance. And this is really frame by frame pose estimation. So there's no post-hoc filtering or other processing, but of course you can do that as you like. All right, so in the ensuing years, this has really been applied to a myriad of contexts. Even going back to Muybridge, I had some fun with this, but anything in cheetahs or fish or mice in different contexts, of course. So it's really species agnostic, it's user defined and really tailorable to your particular applications. The original version has changed significantly since our adaptation of DeepLabCut, of course, which we were inspired by. It was one of the top performing human algorithms on the benchmark at the time. And so now we've, of course, DeepLabCut is built on the open source Python stack. We still continue to innovate and work in these areas actively in our laboratory. We have more than 15 different network types now, which perform state-of-the-art on other tasks. We try to allow users to also build and develop the code as they like and deploy it for large-scale applications. In the past year, we've worked closely with other open source packages like Bonsai and Autopilot to actually build this in such that this could be used in the laboratory with ease. I'm most excited about also is all the great tools that are coming out to do post-processing, right? Because at some level, I've just removed the challenge of doing digitization for you, we and others, royal we. But there's a lot of work in this space. And so I hope Gordon and others will also talk about this and at the workshop on Saturday. But as I alluded to, now there's many toolboxes in this space. And so we wrote a primer piece in Neuron that was just published a few months ago that tries to give you both intuition about how these networks and algorithms actually work. What are the key decisions to make? How do you optimize? How do you do data augmentation? Which networks you should select? What type of options are out there? And obviously you shouldn't necessarily read this text. So I just want to point you to this as a deeper resource for you to try to get some intuition and also discuss the pitfalls and limitations of such tools. All right, but the next really big thing is, say you got poses and what's next. So I'd just like to highlight in the next few slides, like how these types of tools can be used to study movement and movement variability. So as you see, really the top part of this is, of course, any one of these different approaches can be used to extract features or key points, namely the pose. But then you can do things like kinematic analysis, as I'm showing on the top row, very nice example from Oli Keen's lab. In the middle, here's a nice example of using variational autoencoders to try to learn hidden latent spaces, which then can be embedded into, say, things like UMAP spaces to look at, say, how an animal might move around this space. And these embeddings can then be nicely compared, as Gordon Berman and others have shown. And the last example is also a nice example from Robert Johnson, where they used zebrafish in a large platform to study a hunting assay, where then they also used a very similar approach as Motion Mapper, namely a nice wavelet transform and embedding into the t-SNE space. And so we actually have a review forthcoming, particularly for movement and motor control, trying to capture some of these things. But since this is arguably a session on motor control, just digging in a little bit more and thinking about variability, we hope these types of tools will actually allow you to look at this trial by trial availability at much greater scale and having this automated and quantifiable and less, prone to bias and human error. So on the left side, I'm showing two beautiful examples from Sylvia Arbor's lab, where they looked at manipulating brainstem circuits, and you can see the variability in the reaching on the bottom and also on the top across animals. Again, this example from Oli Keen, and in our laboratory, we're very much interested in looking at movements in 3D space, both in natural contexts and in these sort of more constrained task settings, which I'm showing you here with a mouse with a little manipulandum. I think another key point for the future will also be the ability to intervene in motor tasks, right? So just like this nice example of being able to change the floor for being slippery or less slippery, if you can do this in real time in the sense based on, say, a posture of the animal or a particular behavioral feature, I think this is going to be really important. So of course, others have worked in this space, but I know my own work best. So this is a recent paper we just published in eLife a few months ago, where we showed that actually these types of markerless based pose estimation algorithms can be used to do real-time feedback. And indeed, you can even build on modules to do low latency, even zero latency feedback, because many times the time point at which you intervene becomes really crucial, right? A mouse pull is very short, or even say within a stride of a human if you want to intervene. And so the graphs are obviously not, the details don't matter. Everything is at dlclab.deeplabcut.org. But what we want to show you, or just highlight in this slide, is that this can be quite fast, so more than 100 frames per second. We have options for you to do dynamically cropping, also to imbue this onto embedded systems like the NVIDIA Jetsons, and it even works on your MacBook. So if you wanted to download it and play around with your webcam, this would work out of the box for you. And we have some models for you to play with in that sense as well. But I think kind of moving to the other side of things is that, measuring and modeling the motor system and thinking about things like variability and control and learning will also require us to start thinking about how to use machine learning from a different angle. And so really, I could have a whole talk on the history of this, but quite made famous by Jim DiCarlo and Don Yamans, making models of the visual system and really pushed forward as a field by so many people. There's been this great notion of using task-driven modeling. So namely taking in the inputs, if it's vision, right, you can train a network on all the visual input. And what they've shown is that these networks actually start to look like biological things. So we've worked in the space in the proprioceptive system, and others like John Michaels have done beautiful, elegant work on combining sensory motor control. And one idea I just want to put forth is that I think in the future of this, what we'll also be able to do, and we're doing it now, is that you can take, say, motion capture, whether that's marker or markerless, constrain these biophysical models, build task-driven networks, and really start to ask new questions and hypotheses about the system and the agent. So we hope that that's going to be an exciting platform for years to come. So just to wrap up, just some quick take-homes. I hope I've left you with one message, which is I think deep learning has really revolutionized our ability to analyze behavior with precision and at scale. New approaches to modeling the sensory motor system, I believe, will enable new hypotheses and, of course, new tools for things like building interesting control systems and robotics. And lastly, I think, we're at this junction of using machine learning, high data loads, and a lot of new exciting tools. And so I think this is going to be a super powerful platform in the future to test many of the ideas that we're discussing here today. And with that, I'd just like to, of course, thank my funders, particularly the Borrelli Foundation, Chan Zuckerberg, EPFL, NVIDIA, Harvard, and my laboratory. And I'm looking forward to your questions. Thanks very much for the time.
Thank you, Mackenzie. We have a couple questions from the panelists. Tricia, you want to go ahead?
Sure, yeah. I was just wondering, in terms of for human pose estimation, if there are limitations and errors induced by, like with marker motion capture, with clothing, with movement of clothing, with changes in what the person's wearing from one video to another, changes in background, or if they're overground, turning, etc. And then for closed loop real-time feedback, that sounds really exciting. Are there any limitations with like the computational power needed for that? Does that become a factor at all?
Yeah, great questions. So the first one, yes, you're absolutely right. No matter whether it's humans or even mice, which we can think of, they're basically living in this big blubbery fur coat. These are things if you're trying to understand the skeleton, right? So I think, many in the field have done incredible work, say with, I'm most familiar with, say, George Lauder, where they do x-rays and you put beads in the bones and then you get real ground truth. There has been one nice paper actually comparing XROM to DeepLabCut, and they showed it was equally as accurate. So at that point, it becomes how good the human is at labeling, right? So if you're a very messy labeler, then you're going to imbue a lot of ambiguity into your networks. And things like markers also on clothing, they suffer from soft tissue artifacts and movement. So it really just comes down to this question of how accurate do you really need to be, particularly for biomechanical modeling. There's some smart ways to actually constrain your movement from the markers onto, say, the skeleton. And so I think that's a really viable path forward for these more detailed motor control type questions. Yeah. The second part of your question about closed loop feedback, of course, there's always speed accuracy trade-off with both networks that you choose and also the hardware that you have accessible. So what we showed is that on reasonably sized or small images, which again, you can dynamically crop to, say, find your object that you're looking for in your image. I think on, my little wimpy MacBook Pro, this runs at 40 to 50 frames per second, but certainly in high-end GPUs, like a few $1,000s, then you're going to get a quite nice network that you can do this at nice speeds. So I think that's quite exciting. I'm biased, but I think it's a nice way forward. And we can also run, of course, DeepLabCut in real time, but not only that, but then also predict into the future, right? So that's the real kind of computational, advantage with these types of models, is that we can actually predict future movement and then trigger, say, a TTL or any type of intervention that you want to do based on this forward prediction module.
Great, Mackenzie. That's so many exciting advances coming up that I'm really looking forward to. We have several people in the chat, Jill Mitnick-Gray, Alessandro Badura, saying that they've found DeepLabCut to be very helpful in their lab. And so this is really, I think, expanding the ability of people to do this and do it on a budget. The video capture is one of the largest line item budgets in my lab. So there are a couple questions here about whether there are metrics provided about the precision of the estimated points, and whether, I guess you sort of answered it, whether you're getting the pixel of the landmarks or some other 3D metric about the sort of identified point.
Yeah, great questions. And thanks to those of you in the chat for the kind words. As an open source free software developer, it's always really nice to get feedback, because it's always sometimes a thankless job. So it's really appreciated. Yes, great quick question. So you do get pixel coordinates. I'm not going to go through the exact algorithm that we use to find the exact one. You can actually have sub-pixel accuracy with this. And we do have some basic tools for doing this in 3D, right? So you can do that. But also there's been some really nice helper packages essentially developed around DeepLabCut to do this. So we have if you go to our GitHub website, we actually link to everyone else's packages that have that take, say, the outputs of DLC and/or show you how to do this for 3D. But you absolutely can get 3D coordinates out of the main package if you wish. Yeah, maybe I missed one part of your question, Lena, in the middle part there.
I think it's maybe about the precision of the.
Right. Yeah. Mm-hmm. Yeah, sorry, that's great. So yes, evaluation metrics, absolutely. So that's a really crucial point. especially when you have low data sets, right? So the idea is what you do is when you label in the package, then you decide, there's some default values, of course, but you split your data into training and test. And so some images with your ground truth labels are pushed forward to train the network, some are held out. And then when you run evaluates on this, you're actually then pushing these, held out images that the network has never been trained on. And then we literally compute just the Euclidean distance in in pixel space of how good the network is compared to your label, right? And so we've shown that to be as accurate as humans. So human variability, speaking of variability, obviously comes into play with this. And so that's a really important point actually, is measuring your own variability is a crucial thing. We're thinking more and more about adding in tools to allow people to quickly try to assess their variability without having them to label the whole data set twice. So if you guys are interested in such a thing, definitely hit me up on the image forum about this and I'd be happy to chat. But yes, these tools are built in to give you pixel accuracy, and you can convert this to real units in space, like centimeters and millimeters as you like. So.
All right, well, thank you very much, Mackenzie. And with that, we'll move on to our next speaker, Mackenzie, okay, who is Gordon Berman from Emory University, one of my colleagues and collaborators in Emory Physics and Biology.
Great. Can everyone see the screen?
Yes.
Right. Hi. So thanks, Lena, for the introduction. So I'm going to talk a bit about sort of general thoughts I've had about thinking about variability in behavior. This will be a little bit more philosophical talk than you might be used to seeing from me, from those of you who've seen me give a talk before. And this was sort of what I have up on the slide now is sort of my kind of what I had envisioned when I thought about giving this talk, thinking about how do we describe sort of variability in sort of smooth, dynamical form, and trying to understand how do we link things between levels. But as I really over the last couple of weeks, as I started writing this talk, sort of in meme language, this is how it started, and this is sort of how we're going. I think that really what I want to talk about today is sort of what do we talk about when we talk about variability in behavior, with apologies to Raymond Carver, Edward Hopper, and Banksy. So I mean, really trying to think about like what are the things we mean about when we think about variability. This is obviously an extreme example of variability in animal behavior, but the notion is when we're actually trying to quantify these things, it's really how do you link between different levels of representation, and representation of behavior is really going to be a key aspect. And this is something that Mackenzie talked about really nicely in a previous talk. So in general, just an overview, like my group is interested in studying behavior in natural contexts, as opposed to traditionally sort of the more sort of lab-based constrained context that we see. And I think sort of in relation to sort of the beautiful work that Monica showed in the first talk today, what we can see is in a natural environment, there's all sorts of stuff that modulate the gait. So the gait of the dog running in the wild is going to be modulated by the ground running on it, the sensory information coming in, or for example, maybe how tired it is. As any runner knows, when you start getting tired, your gait's going to start changing somewhat. And so the notion of what that would mean as a stereotype behavior compared to the variable behavior is going to change. And so really thinking about variability in these contexts poses new kind of representational challenges of how to describe it, how to quantify the behavior. And sort of the way to think about it, so it's always fun to use the exact same movie that was in the previous talk, but you see from Mackenzie's previous work, you can wind up seeing like behavior can be these things on these really short sort of dynamic timescales, or it could be something slightly longer, looking at locomotion, which involves multiple different aspects of movement. But really what can modulate all of these mechanical movements, which at the end are the things that are being actuated when we talk about behavior, are longer timescale states like circadian rhythm or social dynamics or age or tired, fatigue, hunger. All of these things are going to have subtle effects on the variability that we see, and how to disentangle all of those things together winds up being one of the big problems that we have when we're thinking about variability. And of course, this is something that sort of the classical ethology literature is rife with. So far from the first person to have noticed this. But sort of the difficulty is that it's very difficult to move backwards and forwards in timescales. Right? So people generally tend to think about, we have this behavior, we're going to sort of break it down to a bunch of components, and then maybe we can move those components around, or we can have sort of long time scale things which are modulating the behaviors that an animal performs. But what we don't talk about as much is how do the behaviors that emerge, how do the mechanics of behaviors, the behaviors that are being performing, affect the next behaviors or the internal states of the animal, and accordingly, how do those internal states affect the behavior of the animal, and vice versa. And sort of this being the silly theorist that I am, sort of this is sort of the picture I have in my head. Instead of thinking about behavior in these sort of sets of static behaviors, what I sort of think about it kind of is this landscape, where you have basically a bunch of minima, and maybe we could imagine each of these minima here on this image might be, say, for example, a stereotype behavior like walking or grooming your head. And then what you have is within that behavior, there's some amount of sort of random fluctuations that are going to cause some variability. But then on top of it, this whole landscape is shifting. And so by this landscape shifting, maybe by, say, getting tired, that could cause some slight variations in gait. And sort of, I've written this down as sort of like a Fokker-Planck equation, but in reality, I don't think literally that I want to take this model seriously, but this is sort of the crazy sort of thing that I have in my head. And what I'm trying to do is think about maybe what sort of metaphors can we use to build this sort of moving forward. And I'm not going to give you a particularly great answer, but hopefully there'll be some sort of great discussion on it. So the first thing what we want to talk about is, what do we mean by variability? Right. So the standard way that what people talk about variability is difference in the mean postural trajectory across trials, at least in the terms of biomechanics. Right. So let's say this is some position of some joint angle as a function of time, and I've laid data from a bunch of different trials on top of each other. We can calculate that mean trajectory over all of those trials, and then we can use that to calculate sort of what's that variance around that, or other sort of similar statistics, depending on how we want to quantify that. And this is typically what people mean by variability. But in reality, though, this could be involving a bunch of different factors, as we all know. Right? So I'm going to briefly show an example, and I think Lena might bring this up a little bit later. So this is work that we've been doing in collaboration with Lena and Trisha, as well as a really fantastic grad student, Tanya Winner, so looking at trying to develop low dimensional representations of postural dynamics. So don't have to pay too much attention on exactly how this is created for a moment, but this is a representation of a human's posture as they're walking in an environment. And this is the mean trajectory of each individual, with each different line is a different speed, and each color is a different participant. And so we can see that there's certain variability. Even ignoring that intertrial variability, there's still a fair amount of variability across individuals. But beyond that inter-individual variability, we also have intercondition variability. So on the right, this is the same type of measures, but now looking at individuals who have suffered strokes and are in the process of rehabilitation. And so now we can see that we have this additional extra type of variability that can emerge between what happens not just in the normal functioning condition, but in conditions where we have individuals that are trying to recover their gaits. And so to get further into differences between individuals, right? So now we've moved to humans down to flies, which are sort of where I've sort of spent most of my scientific career, thinking about like, what are the different sort of behavioral repertoires that an animal goes? So that last one was just looking at humans walking on a treadmill. Here, we're going to say we have a fly running around in sort of this existentialist hell in a space without any particular environment around us, and just say measure what are the behaviors that that animal is doing over that time period. And average across an hour, you get a bunch of these different things. So this is this abstract representation that Mackenzie's referenced in her previous talk, where sort of each peak in this space represents a different stereotype behavior, like idle or locomotion or posterior or anterior grooming from a fly. And each one of these different maps is a different individual. And you can see there's a fair amount of variability across individuals, even averaging over a full hour's worth of data. So this was interesting. But then if you look a little bit closer, so now what I'm doing is I'm animating these. So what you can see now is now instead of looking at the full hour, now what we're doing is we're looking at a moving average of 10-minute chunks moving as we going through the data set. And what you can see is this smooth variation between the behaviors that are emerging. And that smooth variation looks a lot like the variability between individuals. So that variability across time, in some ways, is hard to disambiguate between that variability across individuals. So we sort of saw this and sat on it, didn't really say anything much about it. But then, so a few years later, when we started to pick up on a completely separate project, we thought about, so what is the notion about variability across different species of animals? And so here we looked at 6 different species of fruit flies, and we were trying to do a project where we were trying to reconstruct the dynamics of fly behavior across evolution. And so what we could see, and I'm not going to go too much into the detail, but what we could see essentially is, as we tried to reconstruct, so these are sort of the average behaviors across these 6 species, but then if you look at the behaviors of each individual within each of those species, which is sort of displayed here on this low dimensional embedding, where 2 points here are put closer together, if their kind of maps look more similar, we can see that there's a fair amount of variability within a species, but that there's some larger overall structure. And maybe I will skip this, but the general sort of thing that we learned from this is that when we tried to look at that variability within individuals, within sort of an individual species, what we found is actually this interesting duality. And if you're curious, so this is work by Damian Hernandez and Catalina Rivera in particular, what they found was that essentially that variability between individuals is almost identically mathematically to that variability across time. And so trying to actually understand how to disambiguate those sorts of things as we move through winds up being an important set of dynamics. So that gets us back to this sort of overall picture of, we have this underlying sort of representation which might be changing. And how might we, like, what might be a way to move forward? And again, I'm not going to give an answer today, but sort of my overall arching types of thoughts on the issue. And what people typically talk about this is in terms of language and syntax. And you don't have to take my word on it. Like, this is just what I did, is I went on my ReadCube thing and I just looked for syntax and just pulled up a bunch of papers. And you can see sort of there's a bunch of different ways in which sort of syntax and grammar has become a notion for underlying dynamics. However, the problem with syntax is that it winds up, there's no way of moving backwards and forward, right? When we're talking about dynamics, really we're trying to go from that microstructure back to the macrostructure. And how do we move back and forth those things? This winds up being a fairly difficult sort of metaphor that we want to use to build forward. So what might be a better metaphor? And the metaphor that I'm going to sort of talk about, which I think could be useful, might be music, and music theory in particular. I guess I'm just a theorist in all sort of accounts of the word. And so, for example, we can look at long timescale things like a sonata form, or you can look at key signature changes. And I'm running out of time, so I don't want to go belabor this too much, but I just want to give one small example. And so I am a extremely mediocre jazz pianist, and they're thinking about a very simple song programmatically like Autumn Leaves. What you can see is there's underlying chord structures. And I don't have the time to really talk about music theory so much, but the notion is there's underlying chords. These are things like A and D, which are sort of governing what notes could be played. And then on top of that, and there's a natural way in which those underlying chord structures must transition between each other or are allowed to transition between each other. But what's interesting is that you can also get effects like, for example, you have a chord going from a D to a G, where as you have an ascending line, if you play this note, you have to switch the chord at the upper level, otherwise you're getting a tritone. And if people are curious, I could go sort of play what I mean during the question period. And so the hope overall is, what this is pointing out is that there might be some sort of a dynamical framework that we can try and build on top of that, which allows us to go sort of in both directions and allow for variability from both the micro level to the macro level and the macro level to the micro level together, and try and build stuff through. And with that, I will thank all the really great people that have worked on this, the funders, and happy to take any questions.
Thanks, Gordon. That's really great. I really love the music analogy. I've made it before too in some of my writing about muscle synergies, because there's such a large combinatorial problem of like, how do you move? And the music, like we can tell composers by the sort of structure of their music and the chords that they use, even if they have different pieces that are fast or slow. And I'm, having a meta-analysis of those seems like, the right way to go. Although it's really something where I wonder how quantifiable it really is.
Yeah, me too. This is, again, I was asked to be speculative and bold, so I'm being speculative and bold. I mean, I think this is something that we're thinking about. Right.
I think Andrea Davella asks, Gordon, is the behavioral landscape the same for each individual? And are there inter-individual differences in the landscape or in the dynamics within that landscape?
I mean, I think there has to be some degree. I mean, this is always the problem that we're working at when we're trying to build representations, is in order to be able to compare and to sort of aggregate information across individuals, we need to have a common reference frame. And so to a certain extent, we need to put those individuals into the same reference frame. And so in this case, it might be a landscape, but the problem is different individuals might have slightly different rules of governing within that. And how do you kind of build and represent those things winds up always being one of the challenges. And how do we like one of sort of the common jokes in physics is that experimentalists like measuring means and theorists like experimentalists to measure variability or variances. And so I think that winds up being kind of how do you encode that variability is sort of one of these representational questions. And we've done some things on this. Other cases, we just lump everybody together and try and do the best we can. And yeah, it's just going to depend on the specific questions being asked.
Yeah, I mean, I actually, in my talk, it's really weird. I have a language analogy as well, but if you think about different languages, you might suspect that there are different landscapes that are potentially stereotyped across different groups. Trisha, did you have a question?
Yeah, I agree. I love the syntax analogy. Actually, I'm not as much a music person. I love listening to music, but know nothing about music theory. But this example of book, chapter, paragraph, sentence. So in the context of human movements, gait, which we study, it would be interesting to see from one stride to another, you would expect that the nuances of the verb sentence structure analogy should change, right? And the variability at the level of book, chapter, the higher levels might not vary even within someone with stroke before and after. So it would be interesting to see if we can parse out these levels of structural variability and which of them change with time. The whole notion of variability across time, across individual, again, makes it even more complicated. So if nothing else, I think there's just lots of good questions of how do we address that and how do we start unpacking variability at these different levels. And then now do we have too much information and now we have to go back and organize it. So these analogies with music and syntax are really useful there.
I mean, at the end of the day, I think what you have to do is you have to have some sort of prior on how you're going to disentangle these different aspects of variability, because I think even at an instrumental sort of fundamental level, if you're just going to approach this blindly, it's hard to disambiguate these things. You have to have some sort of underlying mechanism or model. And that's sort of where, sort of as scientists, we're going to have to try and earn our money here, is really try and think about the systems that we're looking at and trying to impose those types of priors. And I mean, I think in biomechanics, to me, is always a good sort of starting place for that, because the physics puts some pretty hard constraints about what can be and what can't be done, as Monica very beautifully showed.
Great. So there's a question from, I'm going to get this wrong, Budadita Chowdhury. Do you see similarities with your behavioral landscape to fitness landscapes? And if so, would you imagine the z-axis associated with individual fitness?
I wouldn't say, I mean, so I would say this is a very separate notion than fitness. Fitness, sort of imagine like you might have some sort of fluctuating fitness landscape. And I mean, maybe if I were really to stretch, and this is probably wrong, so I shouldn't say it, but I'll say it anyway, is basically the idea that maybe for to try and locally optimize what that animal is trying to do at a given moment, maybe we could think of that as a sort of a personal fitness landscape for that individual. But to think about that in evolutionary context, I think you would have to say, look at the landscape, and how the animal acts within the environment is actually the thing that's being selected on. And so it's sort of a meta, the selection would be on a meta landscape of the landscape.
So Josh Kashabak says, lovely talk and great operational framework. Reminds me of the idea of hierarchical control. Do you have any insight into the rules and variability that govern transitions between control modes?
Yeah, I mean, so we certainly have looked a lot at that. I mean, and there's certainly a lot of principles. I mean, if we go back to the, if we go back over here to the, like all the classical work in ethology, we can see that there's a lot of work in trying to think, how do you move between these different things? And we see a bunch of different factors, and certain things are obvious, like long time scale, you get circadian rhythms, so you get sort of structure between sort of more awake versus more tired things. But really sort of what I want to know is, you have this large number of set of internal states, and how do they interact with each other? The example I like to give is, if you're in bed at night and hungry, do you decide to get up and eat, or do you decide to sleep? So you have these two sort of internal states of hunger and tiredness that are conflicting with each other. And as animals, we have to figure out how to manage those conflicting sort of internal impulses, and how do you argue? And so I have some sort of thoughts on how the brain, other sort of structures might take this out. But again, those are too speculative even for here. So
great. And last question here. Seems like from Tanya Shea, seems like being able to quantify stereotypy versus variability requires a ton of data. Is there a way to assess this with smaller data sets, typical of vertebrate work, for example?
I would say, so there's a few things. So first of all, I would say that I think there are some recent work, largely thanks to one of our panelists, Mackenzie, on just increasing the amount of data. And that's entirely, like, all of these things that we're talking about are data hungry. And I'm a silly theorist, so when I'm thinking about methods, I tend to start thinking in the worlds of where you have an infinite amount of perfect data, and then sort of working your way backwards towards the world as it actually is. And so there's always a challenge to figure out exactly how much data that one needs to collect to ask a specific question. And of course, like, there are certain questions that we can't ask because they take too much data, but part of sort of what our jobs are is to figure out what are the questions we can ask with the data that's feasible. And if there's a question we really care about, is it feasible to somehow increase our amount of data that we need? But yeah, no, I mean, that's always the question.
That's why we have Mackenzie. Okay, great. With that, we're going to thank you, Gordon. With that, we'll move on to the last speaker of our first session before we take a break. And moving into humans, we have welcome Helsi Torres-Ubieda, who's an associate professor at University of Pittsburgh and currently joining us from Italy. I don't hear you.
Okay. Yeah, I guess, can you hear me now?
Yes.
Very good. Okay. Well, thanks so much, Dina, for the invitation, and Tricia and everyone else, the other organizers. It is my pleasure to present to you this work that we have done in the analysis of muscle activity, and how was particularly related to this topic of variability versus stereotypy. There is this, how the information of variability can tell us something about the motor system of every individual. So I'm going to tell you that in my lab, we study sensory motor adaptation. And by that, I mean that, we do this in the context of locomotion, and we're constantly interacting with different kinds of environments, like, walking on snow, ice, or like sand, for example. And each one of these terrains imposes distinct motor demands that we are able to meet by adapting our movements to these demands. And then at the same time, there are also intrinsic changes, like, for example, growth, or like pregnancy, fatigue. And through these processes of motor adaptation, we're able to adjust our movements to, walk without falling. So in the this question of stereotypy versus variability, I wanted to define what I mean by variability. So both Monica and maybe even Gordon have mostly talked about the variability that occurs when one individual or one animal generates exact same movement multiple times, like across multiple trials. In the case of my talk, I'm going to be mostly talking about the variability that occurs across subjects. And so for example, in these two cases, for this variable A that I am plotting in the y-axis and two different groups of people like control subjects or stroke individuals, you observe that, perhaps the mean is comparable, but there's a lot of inter-subject variability. And what the talk that I'm going to tell, what I'm going to tell you about is how that individual variability could mean something. Okay, so I'm going to be particularly focusing on aging, and we study these using in the laboratory. We study these using a treadmill like the one that I'm showing you here that has two belts that can move at different speeds. So with that type of treadmill, we can generate a novel environment and study this process of motor adaptation. So at the top, what you see is that sometimes the two belts are moving at the same speed, and then all of a sudden, then we can make one belt move faster than the other. So of course, that's a novel situation, and that you will see a corrective response like the one that is shown in there. But then this is a sustained and predictable perturbation that if you maintain over a few minutes, like 15, 20 minutes, what you observe is that even though the two belts are still moving at different speeds, in this particular case, the subject is able to adjust their movements and walk a little bit more smoothly, towards the end of this adaptation period. And now, when you transition back to the legs move at this, moving at the same speed, now what you observe is again this type of corrective response. So we study this process of motor adaptation by analyzing these corrective responses, and what we hypothesize is that the structure of those corrective responses would be informative about the adapted state of the motor system in every individual. So how is that, what is that we mean by that? So let me start by defining corrective responses. So this is work that Pablo Iturralde did in my lab. He's now faculty in Uruguay, University de la Católica over there. And what I want to show you is how, so he defined a corrective response as a change in muscle activity, really simple. So, what is the muscle activity before and what is the muscle activity after this transition in the environment? And that change in muscle activity, that's what we call a corrective response. Now, I'll show you how, two examples of this. So here you have the activity of one muscle on one leg, in this case, the dominant leg. And this is what I'm showing you here is the muscle activity for this particular muscle before and after this transition. So before the transition is in gray, after the transition is in green in this particular example at the top. And at the bottom, you observe, again, in gray before the transition, this will be another muscle in the non-dominant leg, and in green after the transition. So what you observe is we quantify the structure of muscle activity by discretizing the muscle activity over the gait cycle into 12 individual bins, such that we compute the average activity in each one of those bins and the difference, essentially what that change that occurs. So whenever, and that's what is shown in this sort of row at the bottom. So whenever you see red colors, it means that the activity increased in this particular muscle, and whenever you see blue colors, that means that the activity decreased. So we recorded this on 13 muscles on each leg and look at the structure of those 13 muscles. 15 muscles across each leg. So here is an example of what we observed on average in this group of older adults. And what you observe is that the structure of the, I would like to point out that this is coordinated. So every muscle, every row represents the activity of one muscle that is crossing the hip, knee, and ankle, and how it changes over the gait cycle. And what you observe is that there are some muscles that increase activity and some other muscles that decrease activity in some parts of the gait cycle and then increase it in some other parts of the gait cycle. So that's what we observed. But we had this hypothesis that if subjects adopted this split belt gait as the new normal, then we would observe a corrective response just like the one shown here below. And that this, I'm going to refer to this, predicted pattern of activity as delta EMGA. So that's adaptive. On the other hand, if subjects maintained like the tied situation, or when the two belts move at the same speed as the new normal, then the corrective response, the predicted corrective response that we would observe is indicated here in this other board. So qualitatively, what we observe is that, again, on average, most of our group of subjects are what we observe is more similar to what is this adapted state where they adopt the split situation as the new normal. And we have two patterns of activity, so we can compute a regression equation to quantify how much of what we observe is this adaptive-based muscle pattern versus this environment-based muscle pattern. And that is indicated by these coefficients, beta adapt and beta non-adapt. So just by fitting that regression equation, what we observe is that, once again, on average, after a long period of adaptation that's indicated in black here, we observe a lot of adaptive response and not so much of this environment-driven pattern of activity. Whereas after a short period of adaptation, we observe more of the non-adaptive postural response and less of the adaptive one. Okay, so that's as a group. This is what we observed. And now if we fit this regression in every single individual, then what we observe is that those coefficients change as a function of age. So as subjects become older, they express more of this non-adaptive behavior and less of the adaptive one. So in other words, as people get older, they they do not, so they sort of like react to the split belt situation and walk in that particular environment, but as soon as you remove it, they never adopt that as the new normal. Okay, so this was okay, so what is that I've said so far is that aging seems to decline, reduce this ability of the motor system to adapt as people walk in a novel situation. And this is relevant for neurorehabilitation only because people like Amy Bastian or Darcy Riesman or Julia Choi have shown that individuals with hemiparesis can improve their gait by potentially stimulating these sensorimotor adaptation processes. So I'll show you a quick example of that. So here we have a child that had a hemispherectomy, and as a result from that, she walks with a hemiparetic gait. But then after this period of adaptation, what you observe is that she's walking like, much in more symmetric manner. So the environment itself is doing the exact same thing in these two situations, and it's basically whatever the fact that she adopted a new normal that improved the way that she walked. So with the analysis that I showed, the question was like, okay, do individuals that had a hemiparesis, can they sort of update their movement patterns just and adapt a new normal in their term by looking at their muscle activity? And to do this, so Pablo and Digna did this one study where they sort of like tested 15 stroke subjects that had mild to moderate lesions. These are unilateral lesions, and the cerebellum was intact. So what we found is that, once again, on average, the stroke subjects have comparable level of adaptation to those of controls. So I told you that the structure of muscle activity is indicative of the adapted state of the motor system. This ability declines as people get older, and it's not cerebrum dependent. So what we are doing now is that, we are using this information to determine whether this age-related decline is associated to age-related decline in sensory acuity, and whether that motor ability that we characterize with these regressors can predict long-term changes in individuals that had cerebral damage in their gait. So with that, all that I want to say is that by doing this kind of analysis, what we can say, in my view, is that when one does group analysis, you get information about the consistency, whereas when you look at individual variability, you get information about modulators of that effect. Okay, and with that, I want to thank the funding sources, as well as the people that has done this work, like Pablo Iturralde and Digna, as well as Dulce, who's now picking up the torch and doing this, and everyone else in the lab. Thank you.
Great, thank you, Halsey. I really like that distinction between like the modulators. And I'm wondering if like you think age is really the right modulator. We see a lot of variability with age, or is your method going to help us find maybe another metric that could distinguish people with a better number than age?
Yeah, I mean, that's certainly a good point, in the sense that based on this study, it seems that age is one important modulator of this capacity. But we have, like you say, there might be some other factors that influence how much people adjust their movements. And like, for example, what we're working on right now is that, can you predict, after doing this analysis in one session, can you predict what's going to happen after 5 sessions? Is these long-term changes that occur, are they going to be sort of indicated by how much, like, the people's individual ability to adjust their movements in day one?
So Tom Burke says, hi, Halsey, thank you for this very interesting presentation. Do the older adults improve in another area when their adaptive capability decreases? Do they, for instance, prioritize balance over symmetry?
Okay, so if I understand correctly, Tom's question, so he's saying, so let me just say, whether older adults are kind of prioritizing stability as opposed to symmetry.
Whether the if their adaptive capacity decreases, is it because some other capacity improves? Or do you think it's just a generalized decline in flexibility?
Yeah, that's a good point. I don't think that. Yeah, no, it's a good point. I think that once again, what we were characterizing there is how much people were adopting the split situation as the new normal. I'm not certain that there's this idea that there are limited resources, and that older adults might be sort of using all the resources for maintaining their balance, for example, as opposed to adjusting their gait to this new normal. So I understand, sure, that could be a possibility.
Yeah, and I mean, it sounds sort of like you can't teach an old dog new tricks. As I age, I'm kind of grimacing at that idea, but Jill, so related to that, Jill Minute Gray said, would love to see if older adults who participated in sports may be better to adapt. Have you, you have a stratification of age. If you looked at a bunch of people in the same age, I guess you could maybe, have you looked at other modulators there?
Yeah. I mean, that's certainly very valid. Like, in this, and that's also like of a lot of interest, right? Like, we all age differently. So there are some, like, 80-year-olds that are as good at, in terms of the motor system, than, a 40-year-old person, and vice versa. So it's true that there might be, we require more data and tracking the experiences, that the motor experiences that they have had in order to do that kind of analysis. But I agree that, what we think, so I agree that, we age differently, and it's possible that there are some other modulators. Like the one that we're right now investigating mostly is people's ability to detect that there is some change in the environment, and whether that is informative of how much they adjust their movements. So, that's one of the modulators that we're looking at right now.
Great. Jose, did you have a question? Or Pepe, sorry.
Oh, it was just a comment that, and thank you, Jesse, that nice talk on that, there are some changes in the cortical white matter, right, with age, that may account for some of these. Dorsolateral prefrontal cortex, basal ganglia, some other areas. Right? However, as I will present later on, even in the case of chronic stroke patients that clinically are not are stable, there are ways to trigger cortical reorganization and plasticity to improve motor function. So it's, there's always a window of opportunity, and I hope to discuss that with you later on more and the others. Thank you.
Yeah, certainly. Yeah. I don't mean to, when Lina says, like, you cannot teach an old dog new tricks, I think that that's not necessarily accurate. I mean, I think that, there's always this possibility, exercise, for example, like, it's being shown to reverse even some of the aspects in terms of brain degeneration that we observe with aging. So, like, the work of Erickson. So I think that there's always, a possibility for this to, these other factors to change how much people can, like the flexibility in their motor system, for sure.
Great. We're just about out of time. Just one last question from Kat Daniels that I had too. Do you see individual differences in how long the post-split treadmill, post-speed treadmill adaptations last? That is, if people are kind of adopting it as a new normal versus not, can you see it in the temporal evolution of the kinematics?
Yes. So that's definitely, we haven't done that. I think it's a valid, well, my speculation would be that those that are able to adopt a new normal will be also the ones that would take longer to de-adapt that motor pattern. What we're doing right now is, considering the dynamics, would be the rate at which people are updating their movements in their motor patterns would influence the rate of decay. So that's that kind of I think it's of a lot of interest, but also whether, like, the rate at which they adapt might also influence how much they adopt this split situation as the new normal. So once again, I want to emphasize that in this particular study, they were only, like, doing this for 20 minutes. It's always possible that with a longer period of time, older adults maybe reach, the older people might reach a new adapted state with a longer, they need more exposure.
Wonderful. Thank you, Helsi. And with that, there are many more questions in the chat. We can keep everything going there. Right now, we have a brief break, and we will be resuming again with the second half of this workshop at 11:35, which is in about 7 minutes. All right. Thank you.
All right. So we're going to get started again. This is going to be our second session. Thank you again to Lena Ting for moderating the first session. We're going to have more time for a panel discussion at the end. So don't leave after the last talk. Stay on for about 15 minutes of hopefully more lively conversation and debate on all of this work that we've talked about combined. So we had some good lineup of speakers in the morning. For the next 3 talks, we're going to transition more to work on human movements. Some presentations on neural correlates of movement variability, as well as Lena Ting's going to come in and talk about different kinds of motor behavior that she studied in her lab. So this will have implications for neuro rehab as well. So our next speaker is Nidhi Shitapati, and she is at University of Pennsylvania. She's going to talk about her work about predictive power of within and across subject motor variability.
You'll have to stop your share. There we go.
Can you all see my slides?
Yes, we can.
Okay, cool. So, hey everyone, today I'm going to talk about using movement variability to make predictions. And specifically, I'll talk about 3 studies, one in which I'll use movement variability to understand locomotor control and make predictions about control, using variability to make predictions about locomotor adaptation, and also looking at variability and how it can help us predict neuromotor risk. So the first study that I'm going to talk about is how you can infer locomotor control from intra-subject step-to-step variability, by which I mean within a single subject. So human locomotion is usually stable, and the way that stability and the control for human locomotion is understood typically is you have a normal locomotion, you perturb the locomotion, and then you observe how people recover from the perturbation back to their nominal periodic motion. And what you do is you try to infer how the control policy changed in order to, what is the control policy to go from where you are to where you want to be in the states. And this sort of mapping between the state, between how you try to go to a particular desired state and how you change your controls to get there is how people usually infer control. However, even for so-called steady state or unperturbed or externally unperturbed locomotion, there is step-to-step variability, and this should contain some information about how people are recovering from their own sensory motor noise. So what happens is, even if you're trying to move along some periodic limit cycle, you have self-enforced perturbations due to sensory motor noise, and you're presumably changing your controls, such as where you place your foot, how hard you hit the ground, to recover from these self-inflicted perturbations. And I set out to basically infer this default controller in 2 studies. One is in constant speed running and also in sloped walking, but today I'm going to talk about the running study. So what we did is we had people run at different constant speeds for many, many steps. And there we observe variability both in the body states as well as in the controls. In this here, what I'm showing you is the variability in the ground reaction force. And within this variability, we try to infer a controller, and the structure of the controller that we try to infer is the inputs to the controller were the center of mass states and specifically the deviations in the center of mass states from the average or desired center of mass position and velocity. And then we observed how people are modulating their foot placement and their ground reaction forces in response to these deviations. And so we inferred this control policy. And then what we did is we implemented the inferred controller in silico basically by implementing state-based feedback control on a point mass telescoping leg model. So, and then what we did is when we perturb this model, like as you see in this video, it uses this controller inferred from variability to come back to steady state. And in fact, if we if we try to quantify how large of a perturbation this default controller hidden in our variability can recover from, what we find is that this little dark blue dot that you see here, that is sort of the range of variability that was seen in the experiments. And the basin of attraction in silico is much larger than this variability that from which it was inferred. So which is to say that our default controller that we have, in response to presumably our sensory motor noise, is quite robust and can deal with much larger perturbations. Of course, I'm not claiming here that this is exactly the basin of attraction for much larger perturbations in reality, but this is I think what you can infer from the fact that this basin of attraction is much larger than the variability is that default controller is probably quite robust. And indeed, that is kind of what we see when I try to put these findings of controllers inferred from steady state variability in walking and running. What you see is that the control strategies and control policies that we infer are actually quite similar and sometimes identical to what people do when you give them much larger pushes. So for instance, what my model does when I make it, do a step down looks very similar to what say Monica Daly's sort of bird step down experiments show. So what I'm sort of suggesting here is that this default controller to our own noise that we have, we may just, in many cases, we may just be extrapolating that to larger perturbations, or at least we can extrapolate it to larger perturbations. And similarly, I haven't worked on sort of variability derived control in walking, but in my lab, in my PhD lab, some studies have shown that the variability that you derive from steady state control is extrapolated to deal with larger perturbations, and then it becomes nonlinear beyond a certain point, but there is a region for which you can just use this default controller. So the takeaway from this is, of course, that this intra-subject variability in locomotion, or perhaps even in other movements, contains information about control. And I think it would be really interesting to look into, not just variability in terms of the behavior, but also think about what this could say about neural variability and control at a neural level. I think that's a really interesting direction to explore. Also, that this default controller predicts recoveries to much larger perturbations than the natural variability it was derived from. So the next study that I'm going to talk about is learning, or I should say adaptation. I guess adaptation is just fast learning by exploratory motor variability. So the central question I'm exploring with this study is, and this is unpublished, but the preprint is going to be online. I'll show you the link to that. But the central question here is, what principles underlie locomotor adaptation in a new environment? And this ties back to what Halsey spoke about in her study, because I'll be using the same paradigm to explore this. So one sort of popular locomotor adaptation paradigm in humans is the split belt treadmill walking paradigm. And I'm not going to go into too much detail here because Halsey already did, but let me just quickly show you that there's people walking on treadmills and both the belts are at different speeds and each foot is on a different belt. And so they're constrained in this environment and they show these stereotypical adaptation behaviors. What we set out to do is use a single model that can predict both the short and the long timescale adaptations, and without any fits to the behavioral data, by just making assumptions about what we know about how humans adapt. So the hypothesis that we use is that the nervous system cares about stability on the short time scale, and on a longer time scale, the nervous system cares about energy, of course, while maintaining stability. Just to outline what the model looks like, we have this inner loop that is focused on stability. So what we're doing here is we are modulating the controller, keeping track of what the body states are, and then looking at deviations and then using that to the feedback control. And this control is actually it's identical to the default controller that I spoke about in my previous study. So, our hypothesis here is that people have this default controller that's at play that has this stabilizing effect. And then there's an outer loop that is just modifying the parameters of this inner loop controller subject to some objective. And in this case, the objective is to minimize energy over many steps. So what the outer loop is doing is it's a reinforcement learner that is modifying the controller and moving in the direction of lower and lower energy cost. And it's using this by leveraging exploratory noise to explore sort of the neighborhood and estimate the gradients of the energy in that neighborhood. So let me show you what this looks like in an animation. So you have some energy landscape on a normal treadmill, not a split belt treadmill. And then you have some natural variability. And then you're put on a split belt treadmill and there's this new energy landscape and it's now changed. So you're using what we're hypothesizing here is that people are using this natural variability to explore the gradients in the local neighborhood. And then step by step, their stabilizing controller is modified according to the gradients, and they move towards the new the bowl of the new energy landscape. So that's kind of the outline. And remarkably, what we find is that using this, we're able to capture the qualitative trends, both short time scale and long time scale, on the split belt treadmill using this model, again, with no fits to data. The only free parameter in this model is the learning rate. And that is something that we're actively thinking about how we can, model that by incorporating things like memory. So I spoke about split belt because it sort of links nicely with what Halsey spoke about, but I've also used the same model to predict other kinds of adaptation with no modifications to the model assumptions. So what we find is that there are these entrainment experiments that were done by Ahn and Hogan and Ochoa, where they put a sort of periodically perturbing exoskeleton on a person, and people sort of entrain to that exoskeleton's period. And we find that our model does this as well. We're also able to qualitatively explain the kinds of adaptation that you see for mass perturbations, basically adding asymmetries to people and seeing how they adapt to the mass asymmetries. And just because this session is specifically on variability, I did this quick calculation of, if I add, so there's the variability in adaptation we find in our model is really important because it helps us estimate the local gradient, right? But then there's two aspects to this. There's the exploratory sort of, I'm going to call it noise, and then there's also the sensory noise. So the sensory noise is in this context not helpful because it messes with your estimates, whereas the exploratory noise is helpful because it sort of helps you explore the space better. And what really matters for our model is the ratio between these two. So if I fix the exploratory noise and then just increase the sensory noise, you see that I get worse and worse adaptation. So you see that both in the behavioral adaptation and also in the metabolic rate. So you don't see an improvement in the metabolic rate. So the takeaway here is that exploration-based learning of step-to-step controller predicts short and long time scale transients in adaptation. Cool. So finally, I wanted to also talk about inter-subject variability because this is something that I've looked at in my work as well. So what we did here is we used inter-subject variability as a measure of infant neuromotor risk using a data-driven approach. So the way we did that is we curated a database of normative or healthy infant movements, and basically to obtain a sense of the distribution of what healthy human movements look like, and then healthy infant movements look like. And then we're looking at a new infant video and asking the question, what is the probability that this infant's movement was drawn from this healthy distribution? And here I want to emphasize that the distribution is a proxy for this inter-subject variability, right? So there is a distribution among all the ways that all healthy infants move. And the way that we did this is we went on YouTube and got videos of babies, and then we basically took open pose, which is a adult human pose estimation model, and then we did some transfer learning to make it do better on babies, and we found some improvement. And then this is what the tracking looks like on the babies, an example of what it looks like. So we have this high dimensional kinematic data. What we're getting some pre-registered features from it. We had to pre-register. We couldn't do any exploratory stuff because our clinical data set was not high end. And then what we are trying to find is the probability that the movement belongs to the normative data set. And this probability is now a proxy for the neuromotor risk. And when I look at that, what I see is this is the reference database of healthy or normative infants, and they have they're all spread around 0 because that's how we've defined the risk, and then the low, moderate, and high risk deviate from it. And what we can see is that the high risk infants we're able to detect, but we're not able to detect using this approach the low and moderate risks really well. But then the caveat there is that the clinical sort of measure that we use is itself not good at distinguishing between these two. But nonetheless, this shows promise for this sort of approach, shows promise for detecting neuromotor risk. And again, I'd like to emphasize that, the way that we're quantifying risk here is how far away are you from the healthy variability. So that's another way of putting it. So yeah, so the takeaway here is that inter-subject variability in large-scale data allows classifying infant neuromotor risks. So I just want to put up the summary takeaways in a slide to encourage questions, because I've spoken about 3 different studies. And I'm not going to say that again, but I just want to thank my collaborators and other funding sources as well. I'll leave the summary slide up so that people can ask me questions from the earlier part of the talk.
Thank you, Nidhi. That was a great talk. There's a couple of questions in chat. We also have questions from the panel, Lena and Helsi. So I'll start with one chat question first. Do you have any findings on if the data-driven controllers match theoretical optimal controllers? And this is from Peter Gabriel.
Hi, Peter. Yeah, that's a really interesting question. I've not personally worked on that. It's something that I'm really interested in exploring, especially in whole body movements. But my lab mate, Barry Clark, has done some work in this. And unfortunately, I think it's unpublished, but you can look at his dissertation. So his name is Barrett Clark, and he's looked at basically, so this default controller that we have, can we explain it using optimal control, using energy as a measure, and he's able to predict some of these gains that are there in this default step-to-step variability based control using this kind of optimal control model.
And Lena has a question.
Nidhi, and you're walking, really interesting when you increase the sensory noise that the adaptation went down. And I'm wondering if you think that a change in the good variability, this exploratory variability, could somehow compensate for that?
That's a wonderful question. And I think that has a lot of implications for things like rehabilitation, because one hypothesis for, people who are older or people who have disorders is that the sensory noise is higher, right? And so can we just, by making them explore more, help them find the optimum faster? I think that's an open question, and that is what the model will predict, yes. But it is really interesting to ask, empirically how we can do that, and if that will, cause faster adaptation to exoskeletons, for instance, like assistive devices.
And Helsi had a question as well.
Yeah, nice talk, Nidhi. I was wondering about whether you have played a little bit more with this exploratory noise in your model, only because, I don't know, it, based on the work of Natalia Sanchez in, James Finley lab, it seems that people take about, 45 minutes to actually get to those, values that you showed. And in your model, it seems that it's much faster. So I was just wondering whether, it's possible that humans, they just don't explore as much.
Yeah, so that's a great point. Let me just go back quickly to that slide. So I'd like to clarify that, as I had mentioned, the learning rate is actually a free parameter here. So what sort of processes drive or set the learning rate might actually be what explains the slower adaptation. So my model, I can tune the learning rate to be faster or slower, and that really depends on how quickly you're estimating the gradients, or over what time scale you're using the gradients to estimate the local neighborhood. So I think that is still an open question, and it's not sort of, my model doesn't make any assumptions about the speed at which adaptation is happening. And it is a free parameter that we are actively thinking about, what sort of processes might be at play in setting that parameter. So yeah, to clarify, we don't, the time of how quickly it happens is not predicted by the model, but that it happens and how it happens is predicted.
And we just talked about James Finley's lab. So there's a question in the chat by James Finley.
Mhm.
Can you give some insight about your post-adaptation period? It seems longer. The time course of de-adaptation seems longer than what's been done experimentally.
Yeah, so again, both the sort of learning rates, what it looks like will change depending on what I set the learning rate to. The key finding here is that, the shapes are similar, and that there is this fast time scale that is captured and the slow time scale that is captured. So again, what the time scale itself is dependent on the learning rate, and the learning rate is currently just set by us. So there's no sort of underlying biological or theoretical assumptions behind that.
Thank you. And then Helen Huang has a question about part 2 of your talk. Why does the locomotion adaptation only update the feedback controller? And which model did you use to model human locomotion?
Yeah, so we used this point mass telescoping leg model to model human locomotion, which I think is a great sort of parsimonious model because whatever you say, at the end of the day, we are a point mass with the ability to place our foot somewhere and apply a certain force. And what is the question before that? Sorry, I missed the first question. So it was, what was the model? And then the first question was, oh, right, why does it only update the controller? I guess that's kind of one of the key hypotheses here is that we have this default stabilizing controller that's at play. And because it's so robust, maybe that is what you want to use as an inductive bias for learning. And it makes sense, right? Like you don't want to learn over something that can go unstable really easily. And as our other work has shown, this default controller that people have is stable and it is robust to noise. So if I wanted to learn something, it makes sense that I would modify this stable, robust controller that I already have access to.
We have time for maybe one more question. There's a couple in the audience. One's from Rajiv Ranganathan. He's asking, what's the intuition behind the Z-scores and the infant data? Were you seeing some sort of normal distribution, or is it taking something into account? And then Pepe here has a question about slow versus insidious onset adaptations or perturbations. And have you tried to account for that in your model, the rate of perturbation?
Okay, I'll take the first question first. You may have to tell me the second question again.
Sure.
But yeah, so for this one, we're not making any assumptions about the nature of the distribution itself, but what we are assuming, which may or may not be a good assumption, is that the measures are sort of independent to each other. And so that's the extent to which we're making any assumptions about the distribution here. I guess, what was the other question?
I'll actually let Pepe ask. He's on the panel.
Okay, awesome.
Oh, yeah, hi. So in the upper limb adaptation learning literature, there have been, unique differences between slow versus sudden onset perturbations, right? Adaptation to slow versus sudden onset that recruit different parts of the brain and that have that subjects address them in different ways. Have you tried that with your model?
Yeah, so very little, there's no sort of biological underpinnings here. It's just sort of the basic theory here is that there is this stabilizing controller and there's reinforcement learning that happens around it. It's sort of a behavioral level model. We're not making any direct assumptions that link to any neural processes at all, but these are all really interesting future directions, especially if we try to use truly multi-scale biological models as opposed to this sort of abstract behavioral model.
But in terms of the output of the model, like after effects, and time constants and things like that.
Right. Yeah, so the time, I agree with you that there might be some biological processes at play with the setting of the time constants and the setting of the learning rates. And that is something that we haven't explored, which is why it is currently a free parameter in the model. But what we're trying to say is that this sort of framework of having the stabilizing controller and having reinforcement learning around it captures a lot of the sort of qualitative aspects of what happens when people adapt across different kinds of adaptations.
Thank you.
Thank you. Great talk, Nidhi. So our next speaker is actually the person who's just asking the question. So Pepe Contreras Vidal, he's going to talk about his work about neural correlates and implications for BCI.
Thank you. Thank you, the organizer, for putting together this workshop. Very interesting. And I would like to discuss individuality and variability in neural activity. And to do this, I'm going to focus on the framework of brain-machine interfaces. And I think this BMI framework can help elucidate some of the sources of neural variability and how that can be used to explain some or to design better neurorehabilitation protocols. But also it's another way to try to tease apart central versus peripheral components, for example. And so let's start by reviewing briefly, what the brain-machine interface is. And in this case, you can start with different types of source signals, from penetrating electrodes inside the brain to surface electrodes on the scalp. And in my lab, we focus on the electroencephalography or EEG, non-invasive signal, which detects electrical signals of the brain. And here you see a brain cap with 64 channels, which is what normally we use in our experimental protocols using active EEG. So that's the neural interface. Our recording electrode is EEG brain cap. And with that, we acquire, information about brain waves in real time. And then we use that to train a neural decoder, which translates brain activity associated with intent for movement into motor commands to a machine. This decoder can be as simple as a linear model with memory, a Kalman filter, a Wiener filter, or a deep network that is trained on the data. And so a machine could be a prosthetic hand. This is an example of an experiment we conducted a few years ago with amputees. Of course, the participant can plan the movement, can see the object location, the size, the orientation, and he can plan that movement. And through the neural interface, we can capture those signals related to the grasping and use those signals to control the prosthetic hand. And this can happen from day one, and it's closed loop in real time. But the only visual feedback, the only feedback is visual here. So we're now working to add other type of feedback like tactile and somatosensory. And of course, once you perform a movement with the brain-machine interface, there's going to be an action, and the action will lead to sensory error or reward signals, right? If you fail to achieve your target, you're going to get an error signal, and they're going to affect your reward signal as well. And that comprises the feedback that then is processed by the brain and closing the loop. And so the neural interface is recording both afferent and efferent signals that are generated. So the decoder needs to be able to identify motor intent from that mixture. Now, I'm going to describe two experiments. The first one is an open loop, decoding experiment for 3D central hand movement. The second one is a closed loop experiment in a population of chronic stroke participants. So here, we conducted a study where we had participants perform center-out movements to different targets, as you can see in the figure. This was a study conducted by Trent Bradbury at University of Maryland. And what you can see here from these 5 participants is, you look at the mean finger path, there's variability in the path. We see that subject 1 had the more stereotypical path, very straight, and while some of the other participants show more variability. And as they were performing this movement, we were recording EEG using the brain cap, and then we use a simple linear model here shown on the right of the slide to predict, in this case, the hand velocity any time. So S of n implies or represents the sensor EEG at location N over the scalp, and collected time T. And here we are computing voltage differences between the current and a sample in the past K. And we have N number of channels, in this case 64, and we have L number of lags, that's the memory. And so we need to identify the coefficients A and B that allow the linear model to predict the velocity along the different trajectories. And so after training the model and validating the model using cross validation, you can look at what parts of the scalp are more relevant for the prediction of hand movement, both in the scalp and source spaces. So in this graph, 0 means movement on set. So that's the time of the indecision of central movement. And so we look at up to 100 milliseconds prior to the movement on set. And what you see here are the sensor contribution to the prediction in the model, right? So we can see more or less a Gaussian distribution along time, where the highest contribution is between 60 and 70 milliseconds before or prior to the movement on set, which is, in agreement with the corticospinal delay. And we also see that the highest contribution came from sensors in the contralateral sensory motor parts of the brain. And when we did the source analysis using S-Loretta, we saw that precentral postcentral gyrus was very important in the prediction. So physiologically, this makes sense. And so this was, the first demonstration that a non-invasive EEG signal could be used, in fact, to infer 3D hand movements, allowing us to design now brain machine interfaces based on EEG signals. But what I would like to emphasize right now is, the variability, right? So here you see on the left the coefficient of variation in terms of movement time and movement length of the path for all the subjects. And I'm reproducing here the mean finger path for our convenience, and as well as the kurtosis. So as you saw, the subject 1 had the more stereotypical trajectory, very little variability, and that was reflected both in the coefficient of variation for movement time, movement length, and also the kurtosis. Now, if you correlate the movement variability with the degree of decoding accuracy, we found a negative correlation, which we thought was interesting. So, and so we tried to explain, and there are two possible, not mutually exclusive explanations for that negative correlation. One is more technical, the other one is more neural. So the first one is that the increased movement variability could degrade the decoding accuracy of the decoder. And this would be because you have less similar pairs of EEG patterns and kinematic exemplars for the decoder to learn from. Conversely, less movement variability will result in more similar exemplars for training, leading to a better decoder, right? So that could be one way to explain that. Another more neural explanation would be that subjects differ in their ability to perform the task without practice. So this implies that the strength of a priori neural representation for this type of movements will differ. And so these differences could directly relate to the accuracy with which the representations can be extracted. And so these are not mutually exclusive, but these are two potential explanations that will impact, how you train and who can benefit from the BMI. Now, let me go to the next study. In this case, we have a population of chronic stroke patients who did a brain exoskeleton therapy for 4 weeks. And we use movement-related cortical potentials in the delta band between 0.1 and 1 hertz. And the BMI was trained to detect intent for movement. So we have move or rest. And if the BMI detected intent to move, then it will trigger the movement of the assistive exoskeleton, and the subject will be able to perform a target hearing task to provide visual feedback, therefore closing the loop. So this was a study conducted by my student, Nikunj Bhagat, and it's a collaboration with Dr. Gerard Francisco Fontier, our clinical partners, and Marcio Mali from Wright, and who is a mechanical engineer. And basically, we have baseline. So we conducted a battery of clinical assessments on 10 participants with chronic stroke. We did it twice, 30 days apart, to ensure that they were stable and that any change due to the therapy will not be just to a spontaneous recovery. And after that, we have a session for calibrating the BMI. And after that, we started the therapy. They received 12 sessions of BMI exoskeleton therapy. As you can see on the figure, then we have assessments immediately after the treatment, 2 weeks after, and 2 months after. And so the experimental protocol basically was an EEG-based BMI to monitor and detect movement intent in chronic stroke survivors during the therapy. And the therapy focused on motor imagery of elbow flexion extension via visual feedback in this target hearing task. And the robot assistant was provided by the MAHI upper limb exoskeleton that MAHI developed. And it was initiated only once the BMI detected intent from brain activity of the participant. Now, this is a summary of BMI performance and clinical outcomes. On the left, we have the longitudinal BMI performance. We have the mean group, and also we have 2 exemplars, the best participant and P9 and the worst P7. You can see that the BMI accuracy improved as participants were trained on this protocol, but also the detection time, how soon the BMI was able to recognize the intent change over the sessions. In fact, was reduced. At the beginning during calibration, it was about 500 milliseconds, and after 12 sessions, it was a few milliseconds before we could detect the intent and drive the assisted robots. Now, if we look at the clinical outcomes, we use the Fogelmeyer upper limb assessment and also the action research arm test. And you can see that we found gains immediately post-therapy at 2 weeks, and these gains were maintained at 2 months. Remember, these were chronic patients that were tested twice before the session, so we believe that this improvement, these gains were due to the therapy. The BMI triggering cortical plasticity, and by closing the loop, allowing the brain to learn. This was also seen in the RF test. Now, if we look at individual patients, we look at the minimal clinical important difference. And in the patients, group, we had 2 groups. We had a low functioning group and a high functioning group. And so if we look, for example, at the RF score, the MCID will be 5.7 in this case. And post-treatment, we found that the less affected, mild to modest participants really show gains, while the most affected didn't. If we look at 2 weeks, these gains were maintained, and to our surprise, these gains also were seen after 2 months post-treatment, which was great news for us and for the participants. And just to give you an example of the assessments, participants 2, 4, and 9, as you can see, they have other scores in the single digits, 4, 4, and 9, versus the other ones, and also in the Fugel-Meyer, 26, 21, and 20. So they were severely affected, and they didn't seem to benefit from the intervention. But remember, this is just 4 weeks, so we cannot rule out that this intervention could help if we have more time. Now, this is an example of one participant, a baseline, and then after 2 weeks and 2 months. So you can see from the video that the participant improved considerably in his ability to reach and grab these objects. Just after 4 weeks of training, and with the gains persistent after 2 months. We are currently conducting a longer clinical trial that will have initial component at the clinic, and then we will send the participants to their homes for a longer period of time to really assess both the performance gains and also the cortical change. And so just to conclude, cortical reorganization and motor recovery. So this study suggests that EEG-based BMI can lead to changes in cortical activity in people with chronic stroke after 4 weeks of training. We found that the changes in the amplitude of movement-related cortical potentials in the contralateral EEG electrodes was highly correlated with the functional assessment scores. However, these cortical changes change across participants. So there was variability in neural activity and neural representation, probably in part due to the brain injury. And I want to emphasize that we only trained the elbow, right? They were using the elbow to hit the target, but the improvement were seen also at the wrist and the hand, and as measured with the Fugel-Meyer upper extremity assessment. This may be in part due to an enhanced sensory-motor feedback during the neuro rehab therapy, and also that this gain seems to persist at least after 2 months post treatment. So with that, I'm really happy to answer any questions. Thank you.
Thank you, Pepe. This was a great talk. We already have some questions in chat. Dan
Great.
Song and Scott Bobinger both ask the same question. Have you used pre and post movement beta oscillations as an input to the decoder?
We use, let me see if I understand the question. So we use, we predict the time series, right? For movement in the experiment 1, decoding the actual hand velocity across space-time. And in the second experiment, we detected the intent for movement, right? So, and these were obtained from a subset of electrodes over sensory-motor cortexes in both hemispheres that maximize the prediction. And so we didn't went for a specific brain oscillation, something like that. We were looking more at the readiness potential, the movement-related cortical potential. I hope that answers your question.
Yeah. There's also a question, I think Lena Ting had asked about, do different people need different decoders going with the variability theme? And George Wittenberg has a related question. Was there any intention to stratify or fractionate participants based on their baseline impairment, based on where they started from?
No. Again, we had 3 subjects that were severely affected. They didn't show significant improvement in these 4 weeks of BMI training, but the others did, and they persisted for 2 months. This was, the first study of its kind, I believe, and so now we understand better, the inclusion exclusion criteria we should use in the future. So we were able to optimize the therapy better. Was any control for the BCI after the training? Well, we collected training data, both movement of the course on the screen and EEG over a couple of days to capture context, from day to day, your motivation level, your fatigue level, there are many things that change from day to day, and we wanted to capture context. I think this is very important for BMI. And so we aggregated data from a couple of days to train the decoder. But once the decoder was trained, it was fixed and it didn't change during the intervention. So that's the calibration part. And this has to be done for each participant. Each participant is different, particularly here where they have different type of brain lesions, right? I mean, they are stroke, but hemorrhagic and different subcortical, cortical. So it's really personalized medicine, in that way.
Sounds good.
Any other
Thank
questions?
you. I think these are good for now, but we can come back to it in the panel discussion in 10 minutes. Thank you again. Our next speaker is, so we're coming back full circle to our moderator and co-organizer, Dr. Lena Ting.
Great, thank you, Tricia. It's my pleasure to be speaking about this topic of individual variability, and it's sort of a trajectory of research that was somewhat unexpected, but now thinking back of it on it has really been driven by sort of the diversity of experience I've had in my life as a sort of multicultural, multilingual individual, as well as the diversity of the individuals working in my lab and the diversity of the participants we've had in our lab. So one of the things that I've just really been interested in is why is it that we can identify people by the way they walk? And this was extremely salient to me when as a young college graduate, feeling sort of as an outsider, went to China to try and find a place where I belonged. And it was very clear immediately that people could recognize me from very far away as having a movement pattern that was foreign. And so that would be, hit home that the differences in gait aren't just biomechanical, they reflect our physical beings, our emotional and social experiences. And there isn't a lot of literature on this, but I want to point out this work where Nico Troje took videos of these markers of people walking into kinematic decomposition and was able to demonstrate these sort of axes of coordination across the points in which we're able to perceive different aspects of this movement, if someone is male or female or happy or sad. And what you'll notice here is the point cloud, sort of the points get closer and further away from each other. So these sort of violate biomechanical principles, but it demonstrates sort of how our brains are able to see these differences. And then Karen Liu then implemented in simulation the ability to synthesize these movements that express emotion and individual styles based on preferred patterns of joint torque, which further shows me that these constraints that we see are not, although biomechanics plays a role, it's above and beyond the biomechanics. And that's where I think the role of neural controls and our experiences are extremely important. Now, sort of parallel to Gordon, I want to take a segue here and talk about how we understand speech accents, the structure and the consistent variations there, because the native language biases how we both produce and perceive sounds. So Patricia Kuhl has been studying this in babies for a long time. And what you'll see here is a representation of formant space. So this is the first harmonic frequency and the second harmonic frequency that we hear. And this maps out the sounds of vowels that we can discriminate. And infants are sort of have a blank slate, and they can, each of these regions corresponds to one vowel sound that they can distinguish. And as we learn to speak languages, these boundaries change depending on the number of vowels that there exist in different languages. So what you see here is that in Swedish, they have many more vowels than they do in Japanese. And so what this does to this sort of map of how we interpret sort of this continuous range of formant space is that when we hear sounds that are, somewhere in here, a Japanese person might not be able to distinguish these two vowels that are produced in English because they're overlapping with this single space. And so based on our experience of what's useful to us, we've sort of chunked this very physical and objective space into these categories that correspond to sort of meaningful things. And in fact, it obscures our ability to make certain perceptual distinctions. So this is sort of a model that I use to understand the sort of muscle synergies that many of you, or motor modules that many of you may be familiar with, that we've been working on for many years, and the fact that these motor modules or spatial patterns of muscle activity across the body may vary with experience, training, disease, and rehabilitation. So the idea is that you coordinate multi-muscle patterns, and they're modulated over time here. And like the speech phonemes, these motor modules are like the syllables. I'm not talking about the higher level of words or grammar or chapters of a book that Gordon talked about, but I think that all of those do exist. And one of the challenges is to recognize that hierarchy. So these different modules correspond to these functions that need to occur in biomechanics. And in our early studies with Helsi here, she actually identified these individual differences. And one of the key ideas was she brought in her teammates from her Taekwondo team. So don't get in a fight with Helsi. And we saw that they had this sort of knee bending strategy that was separate from the hip and ankle strategies that we normally see, sort of opening up another degree of freedom that they could use to maintain their balance on our platform. And after that, we worked with Dave Clark as well as Trisha Kazar to show that in stroke, you have the opposite happening. You have a merging of modules and a decrease in the availability of these sort of library of actions that correspond to the level of impairment in people with stroke, and that can affect whether or not a particular rehabilitation is effective or not. I'll talk to you a little bit about the training in ballet dancing in the next slide. And I just wanted to point out that the ways in which these modules change seem to be different across different neurological disorders, which have a lot of implications for the types of rehabilitation that might be needed, as well as metrics of evaluating them. So in the ballet training, this really shed some light on why we think people exhibit these motor accents based on their experience. So in the ballet dancers, we had them walk on a narrow beam, and compared to the novices who we had coming in, they appeared to be more efficient in their movement. So the modules that they used had fewer numbers of muscles in them. And they used the same modules when they were doing overground walking, as well as this very difficult beam walking task, which suggests that the training sort of expands the range of tasks that you can feasibly perform with the motor modules for walking. And because of that, it actually alters the sort of way in which you walk in overground walking, which is a very unconstrained task. And so we would expect that, and this is really good for rehabilitation, meaning that if we train in one context, that it should carry over to another context. Now, the thing that's really annoying to me is that when we look at the kinematics of these movements and we plot them in these standard biomechanical plots, we don't really see a difference. Experts and novices, when they're walking on the beam, look more similar to each other than to themselves walking overground and on the beam. And so something about our the way in which we look at biomechanical measures is obscuring these things that are very easy for us to see by eye. And as Gordon alluded to, Daniel Winner, a PhD student in my lab, wanted to tackle this with machine learning, went out and got 2 extra advisors and started training kinematic data on a recurrent neural network. And what this does, it really captures the dynamics of the kinematics, that is the transitions from one time point to the other. And then we take the guts of this model and identify the sort of low dimensional dynamics within the representation. And finally, with that, the intent is then to generate a gait signature, which we can map out. And these are all the stroke participants, and these are the able-bodied participants. Now, you've already seen the picture that, that Daniel generated for able-bodied individuals walking, this heart-shaped thing. And each of these colors corresponds to one of the gait phases. So during walking, you sort of go around this trajectory. And what you're seeing here is actually a variety of individuals here now colored with a different color, walking at 6 different gait speeds. So you can see that these patterns are relatively invariant across speed, which is interesting because typically the kinematic and kinetic characteristics vary across speeds and cannot be directly compared. In stroke individuals, here are the patterns that we see, and you can see there's a very high range of variability, and it differs for each person. And again, when we look at each person, their trajectories are very consistent across different strides, and actually the range of speeds here was quite different because people's ability to walk was very different. So that's what I'm going to show you right now, and look forward to more work here. Finally, you know, this is a nice descriptor, but I'm really interested in what the underlying mechanisms are that cause these variations. And so what we have done is started looking at brain activity during movement using EEG during balance perturbations, in which it's simpler, easier for us to look at the hierarchical control, sensory motor control mechanisms, right? So we know that when you get a perturbation, you evoke a spinal cord response. We've spent many years looking at this brainstem mediated response, but we know that the brain can become involved in this, what's typically an automatic task. And by looking at the activity and how it spreads across the cortex, we can actually see that there are quite a number of differences between young adults as balance perturbation magnitude increases, older adults where it moves more frontally, and stroke survivors and where it's sort of spreading all over the intact hemisphere. And these variations that we see, you know, we hope tell us something about the capabilities of that individual. And so Aidan Payne, Nina Gosin in my lab have demonstrated that this, this cortical evoked response right at the midline right here, so this is the perturbation onset, the magnitude of this response while they're standing on the platform is larger in people who have more difficulty traversing the beam. So the idea is their automatic brainstem controlled balance is not adequate for this, and they start recruiting cortical resources in order to help them achieve this task. And those who are able to go all the way across the beam seem to have very much smaller levels of cortical signals evoked by this perturbation. Jackie Palmer and Michael Boric collaborated with us to now take this to older adults, where we kind of see the opposite, where the beta coherence, so the connectivity around the sensory motor region is greater in older adults with better balance, as measured by this clinical mini best test, meaning that they're actually using their cortical resources to improve their balance. But in those people where we see it really moving frontally, where they have connectivity between the prefrontal and the sensory motor regions, they have very poor dual task interference, which means that basically they can't walk and talk at the same time. If they start talking, they have to stop walking. And this is an indicator for fall risk. So we're hoping that these sort of brain biomarkers give us another metric by which to measure people's balance ability that could be useful for training and rehabilitation or BMIs or anything like that. So in closing, I want to just say that, you know, everybody's different and we can use it to our advantage for studying principles of movement. And, you know, the first thing I want to advocate is to, you know, embrace variability in your population, your behaviors, and I really eschew averaging your data. And, to remember that the brain can solve the same problem in many different ways through the variations at many levels of cells, circuits, behaviors, and across individuals. And the key, and to remember that the behaviors are often more consistent than the underlying neuromotor activity. So the challenge here is to say, well, what is the structure underlying that variability? So we get information out of it, and how do they, how are they individual specific, so that we see these consistent biases, or what I would call motor accents. That's what I said, define organizing principles. And to do that, in order to do that and embrace this diversity, I've had to collaborate with people who have many different backgrounds than me, sort of lending multiple insights onto the same problem. And I advocate that. All right, so with that, I will end my talk and bring us into a question session.
Thank you, Lena. We already have some questions in chat. There's a question from Peter Gabriel about the ballet dancers data that you presented. Do you think that the reduced number of muscles is optimized to reduce variability in certain directions of movement? Do more muscles create more risk for variability? Would you have comments about that?
So let me see if I understand that question. We think that the reduced number of muscles, so oftentimes there's a low level of muscle activity in many muscles. And so reducing that sort of reduces the amount of co-contraction, would increase the efficiency of the movement, which would be really important in ballet dancers, but perhaps at the cost of stability. And so you would be able to do that if you have a very refined sensory motor control system that can accurately direct your movements. Did I answer that question?
Yep. What you just said also addresses another comment by Tanya about energetics and fewer muscles being normal movement, and how that affects energetics and energy cost. There is
right. Well, so just to say that in our Parkinsonian individuals, after they go through an adaptive tango rehabilitation, we saw muscle synergies, but also more co-activation. And it might be in that case, in order to achieve that consistency, they're actually having to adopt a less energetically efficient strategy.
Andrea also has a question about the PCs that were shown in the plot from Danielle's work. What are those principal components?
So those principal components correspond to, actually Gordon can answer this question, the weightings. So within the internal variables of the network, we can then look at the weightings on those structures that are used to reproduce the walking pattern. And so there's many hundreds of variables there, and we do a dimensional reduction on those internal weightings that characterize a particular person, and then plot them in phase space.
Lena Halsey has a question for you as well on the panel.
Hi, Lina. Thanks for the talk and showing my picture when I was young. So I want to ask about, precisely gait automaticity and how with aging goes down, or you were showing how, like, the cortical activity going up. I was just wondering if you think that can be trained out, like.
So we hope so. We don't know. I think one of the issues is that we simply don't know when people start to engage their cortex. So what we're showing is people with good balance are engaging their cortex, and most of the clinical tests are dealing with prefrontal activity, in which case it might be too late. So most of the tasks are dual task, where you have to count backwards while you're walking. And so we showed that means that you're engaging too frontally, and it's possible that we need to catch people when they start engaging their sensory motor cortices in order to do the training.
Pepe had a question as well. You can go ahead, Pepe.
Yeah, I think it might be interesting to assess cortical efficiency on the ballet dancers. For example, in terms of cortical communication, what's the functional network? I would expect them to show a more focal activation, the They don't think about it, they just perform because it's ingrained in memory and things like that.
That would be really interesting. We just not that long ago started our EEG work, and you know, we may have to go back and see if some of our ballet dancers from the Atlanta Ballet would be willing to come back in, but certainly we see more focal activity in the younger adults compared to the older adults, and then compared to stroke survivors.
We had done a study with dancers as well in Frontiers in Human Neuroscience. I can send it to you.
Great, thanks. I'll have to take a look at that.
There's maybe time for one more question. Rajiv Ranganathan asks if you compared your brain measure predictions for fall risk with simpler behavioral scales or clinical scales like self-efficacy or fear of falling.
We haven't done those. Mostly right now we've done the mini best test, which has some validity for a fall, as well as dual task interference. And this was done with the timed up and go test, where you take the amount of time it takes them to rise from chair, have them do it again while they're counting backwards by, I don't remember if it's 3s or 7s. And the difference in the amount of time there is actually related to the amount of sort of connectivity. And this is electrode based. So something near the front and something near the center. So that is definitely our intention it's actually surprising how much correlation we're seeing between this very general behavioral metric and this sort of more specific brain metric. We were quite surprised.
Okay, so I think we can transition to the group or the panel discussion.
You want me to share the slide?
Sure, that would be great. Yeah, Lena has a slide of questions that were already sent by registrants. And then I was keeping notes as well. There's lots of broad questions about quantifying movement variability and stereotypy, and we talked about some of this. I think one that I'd love to hear the panel's input on and spans across all these talks is this notion of, is variability beneficial or harmful? How do we know what's the optimal level of variability? Too little or too bad can have negative effects on movement. And if you have comments or previous experiments or just, you know, theories on that.
This is open to the panel. Please jump in.
I think it just partially depends. Like the first thing that came to my mind was thinking about songbirds, where in their variability, they have sort of almost two distinct forms. You have your undirected song, which is a lot more variable than their directed song. And so it depends on context and whether you're trying to learn or whether you're trying to perform a rote task. Given the music theory analogies I had earlier, certainly if I'm trying to play a piece I know very well and I'm thinking about it, I do quite poorly. and so that stereotypy is quite beneficial there. But when I'm trying to actually figure out like how to improvise over a particular chord structure, that having that variability is useful. And so I think a lot of it just has to do with this sort of standard explore, exploit type of optimal principles.
Anyone else have comments on that?
Well, I think it's hard, I think we maybe are looking for a one size fits all answer to variability, and I don't think that will exist. We know that variability is one of the tenets of sort of the success of adaptation and evolution, is that we should have variability across individuals in many levels, because the world is unpredictable and changes. And the degree to which, we express our variability isn't just about ourselves, but about, the success of our species. There's some people who are gonna, jump in to a new, eat that berry and they might die. And other people are gonna watch them die and not eat it. But we need both.
Yeah. And I think this was said actually before in chat by Kirti Krishnan after Gordon's talk, that it sounds like we don't even have the language or terminology to capture variability and its different components and structures. And that would be a good start. What's the normal level of variability? How does it change with demands of the task, context, stages of learning, pathology, right? And then Gordon brought up psychosocial, motivation, fatigue factors. So there's just a lot of complexity that's encoded in there, and we don't have good terminology. Maybe we don't have good ways to parse that out. So I guess lots of future directions. Our work is cut out in that way.
I see Andrea there asking a question of stereotypy, just an artifact of poor sampling on the continuum of individual solutions. And I'm not sure about that. I think that there are principles, there are physical principles governing how we move. So when we walk, we have balance, we have gravity, we have mechanics. And so especially in movement, there are particular constraints that we really can't get over, but within the allowable variability, we see it. And when we get to, and I think structure is necessary for, like, maybe I'll call it creativity, generative aspects of movement as well as thoughts, right? So language has a structure, there's stereotype, but there's variability in it that allows us to create new sentences and things like that. So I think that the stereotypy is actually a necessary part, and that variability within the structure allows for building up things modularly. Gordon, maybe you have a better way of saying it.
I think there's, when you thinking about stereotypy, I do think that it's, you have these particular behaviors that tend to be performed more often than you would naively expect if you were wandering through this whole kind of continuum of possible solutions. And some of these, as Lena said, are encoded by the physics. Some are encoded just by there are particularly good solutions to particular problems. And I think overall, it's trying to isolate what stereotypy is this is why sort of I showed in my talk, like, that landscape picture of that. It's sort of this weird kind of not quite tangible topic of, like, if I were to sample through the space, there's some areas where sort of it's easier to get to based off of the neural and physical constraints that are on the behavioral performance, but that doesn't necessarily mean we should take the notion too literally.
Yeah, I'd like to elaborate on that. Like, I think in response to Andrea's question, I think the sort of landscape plot that Gordon showed is a really good one, because there's different kinds of stereotypy that you can see arising in that landscape, right? Some are because there's this nice optimal solution and people are doing that, and that's the stereotypy. Sometimes it's what Lena said, and it's these biomechanical constraints that don't allow you to really do anything else. And so it's maybe kind of quite flat there, the space of allowable solutions. And you can't really talk about optimality, and it interplays really well with variability also. Like, if you have a lot of exploration in that space, then you might find a better solution. And I guess it's an open question whether or not people do that, because there's examples in the literature both for people finding a new optimal solution by exploring the space, and also for not finding. And I think it will be really interesting going forward to see if we can start explaining when people do search and find better solutions and when they don't.
Yeah. There's also Nidhi's talk, you talked about your hypotheses about stability versus energetics and the two loops that are trying to use those as cost functions. What about when there's competing priorities, when, like, in neuropathology, you have to prioritize one over the other and you can't optimize both? There's a question in chat right now from Aiche Lee about trade-off between optimization, energy cost, and variability. So we were talking about variability and stereotypy. You talked about the energetics versus stability. So how do you think we tackle that? How do these different behaviors optimize that as the demands change, as pathology causes, more prioritization for stability, maybe in the case of stroke or older people?
So let me take that first, because you mentioned my talk. So to clarify, I don't use stability as an objective. I use it as sort of a not constraint, but a sort of inductive bias for learning. So you learn along the stabilizing controller. But having said that, I can imagine, it's showing up as an objective in cases where, especially in pathological cases where it's maybe hard to be stable, right? And so, so there it can show up as a very key objective. Whereas, for us, for a lot of locomotions, like for split belt walking and such, it'll be very easy for us to walk stably, or it almost feels trivial. So I agree that it can show up as an objective, and it's really interesting to see how it shows up as an objective in those populations.
Does anyone have comments about how do we best measure variability, and especially in response to interventions or exoskeleton devices, pharmacological treatments, changes in movement patterns? I think Pepe had a comment about that.
I'll try. I think, we all know about variability and the standard kind of mean and standard deviation type of idea. And I think that's really not been adequate for us to understand the variability. And this is where the sort of dimensional reduction types of techniques kind of tell us patterns that underlying that variability, so that the variability is really not random. It is directed, it has some structure. So if I have 10 muscles, they're not all 10 varying independently. And so that's one of the ways in which we've handled it. The other is we actually model the feedback control mechanism and show that many of the variations across individuals is just a matter of changing the sensitivity to a particular sensory signal up and down. And so once we get it into a space where we can understand the structure, we can then start talking meaningfully about the variability and its relationship to function. And this, unfortunately, is not something that I think is just going to pop out of some machine learning model, as Gordon said. And it really, we have to understand deeply the kind of what is the behavior, what are the important aspects of behavior, what is all the information being conveyed by the behavior, such as when I talked about walking, it's not just about getting from place A to B. It's about expressing confidence, about fidelity with an in-group, emotions. All of these things come out that we would count as variability when people come into the lab, and we just average it. So also experimental manipulations, as Monica said, to highlight particular aspects of the variability is also important.
I have a comment on the, I think how devices may affect individuality. I think that was the question. Something that become very interesting for us is to think not only about movement, particularly in the context of BMI, where we are trying always to predict movement intention and then use machines to restore that. But I think it's important not to forget that we also communicate through movement, and part of the variability might be due to that. So that's one reason we started to work with dancers and children and other populations, because we want to account for that contribution, and how that will affect the movement pattern that we are trying to decipher from brain activity. And so I think that might be a source of variability, right? This, the expressive movement component.
Yeah.
And therefore, you use machine like exoskeleton that will affect that part.
True. Yeah. I think Jing has another question for the panel.
Jing, can you? All right, well, let me just read it for her. Yeah. So she says, it seems that we can assume that humans have larger variability, which is possibly a source for learning and creativity. So evolutionarily, where does this variability come from? And what is the neural source of variability?
Hi, so yeah, I have a question. So from all these talks, it's a fascinating talk. Can I?
Yep, we can hear you.
Hear me or not?
Yes, we hear you now. All right, well, let me just read it.
Yeah, okay.
Oh, you'll have to mute your YouTube.
Yeah, I think the evolutionary source of variability, that's a good one. I do not know the answer to that. I'll let other people chime in. It's a good question.
Doesn't Monica or Gordon have something to say about this?
I think that, Lena, you hit the nail on the head earlier when you were talking about how you need variability among individuals in natural populations to be able to be adaptable to allow adaptation to different changes in the environment or novel conditions. So as you say, you encounter a new type of berry, some individuals will go for it straight away, some individuals be more cautious. Having that variability allows for adaptability in natural populations too. So while it's a source of creativity and expression in humans, it's also an important source of adaptability in natural populations.
So are you saying it's genetics or epigenetics?
I think that there's both genetics and epigenetic factors at play. It's not my area of expertise, but early life experiences can have long-term influence on brain function and personality, certainly. So, and then there's also factors that can be epigenetic in terms of traumatic events influencing brain function in offspring, for example. So if there's a sudden change in environment and this causes, like negative life experiences, then that can actually be passed on to to children and cause them to be more risk averse and behave in ways that might be adaptive under those circumstances.
Yes, absolutely. And going back to my language thing, the reason I'm really just fond of the accents is that I actually spoke 3 languages before the age of 5, which is, we know the critical period is by 7. And so after that, I learned to speak French, and I have apparently quite a good accent because I can hear different sounds that I wouldn't if I was monolingual. So I agree that this early life experience really shapes how our brains attribute meaning to the experiences that we have. So based on language, and that will happen. It's a good analogy, but it has to be happening in all aspects of the things that we learn. And that's the important part, is the language phonemes. It's a learned behavior.
Yep. Good discussion. I think the last maybe that we have a couple of minutes for is things to think about moving forward for the field. How do we account for this in our studies better? Are there important research questions you'd like to make a quick comment on?
One thing I'll mention that sort of I think ties a lot of this together is that what we think a lot about in our group is how to disentangle all of these different sources of variability, which in some cases are at a theoretical level not disentangleable. And so what we need to do is be able to figure out how to incorporate the knowledge of the specific systems we're focusing on, or what connections we can make across different systems, and what connections we can't make in order to try and sort of disentangle what these sources are and figure out what things we even need to bother with disentangling. And so those are sort of kind of the things that we're trying to develop in terms of on the methodological side.
And quick follow-up of that, you talked about motivation and other factors. Do you think we need more physiological monitoring, or do you think a lot of these other factors are encoded in the behavior? Do we have the computational tools to parse it out without having to go in and measure EEG and sympathetic nervous system activity and like all the physiological signals separately?
I mean, I think there's what comes to my mind is back before he was Richard Dawkins, when Richard Dawkins was a expert in fly grooming behavior. He has this lovely sort of thinking about kind of the neurophysiologist's nirvana, where you knew the firing of every single synapse and the connection of across a whole animal, and sort of that hardware interpretation. But knowing that hardware, it'd be really hard to figure out exactly kind of what are the general principles across different organisms. And so really what behavior gives us is the software in a way. It gives us how are the ways that animals are moving, and then from that software we can hopefully try and make constraints on what the hardware might look like, and then make predictions. And hopefully the two approaches kind of meet each other in the middle at some point, and we're able to try and sort of more formally make these links.
I don't know. I think Kelsey's showed that when their individuals adapt these different strategies, their kinematics look pretty similar. Is that right, Kelsey?
Yeah, I mean, I did like sort of the summary or take-home message slide from Lina, where she says, like, as a researcher, go beyond the average, right? And just take into account within subject, trial to trial changes, as well as across individuals. And that we just need to push ourselves and our trainees to try to investigate into like the mechanisms underlying those subject specific or individual changes.
I think for me, like, we do our power analyses based on the mean or standard deviation of discriminating groups in our grants. And in reality, what we end up doing is needing to do more of a association or correlation analysis. And, basically it means that for that power analysis, we have to increase our numbers to probably about 35 and make sure that people are stratified across whatever independent variable we think is driving that variability. So it does require us to shift our scientific habits away from means.
One comment in terms of designing BMI systems, I think it's very important to record movement and brain activity in action and in context. I mean, this is important. Outside the lab, we tend to limit subjects, and that's not naturally what happens. The other is that we need to do more integration of computational studies with experimental approaches. I think that from the computational, you can come with some prediction that you can test and close the loop and so on. So those are my two recommendations. Data sharing will be another one, so that we can remove these limits on the number of people we can test.
Yes, agreed. Then Gordon can have his infinite high quality data. then we can actually be closer to reality. Yeah,
I knew I'd get in trouble for that.
I think we're running out of time, so I'll let Lina just quickly close down. I certainly want to say thank you to all the speakers and to TMLS, the organizers, especially Tiara Ward and Amanda Scott. And I'll let Lina have the last word. She's the organizer here.
Well, I'm just so pleased to see all of our friends and colleagues around the world able to participate in this event. And we hope that we can do this in person one day, but also allow it to be accessible by all of you. Thank you to all of our wonderful speakers from across the US and in Europe right now, and sharing your insights and the sort of push towards the next stage, next level of understanding diversity. Thank you.
Thank you all. Bye-bye.
Thank you. Bye-bye, everyone. Have a good rest of the day.
Thank you.
Bye. Thank you.
Awesome. Thanks so much.